I'm checking the sound here. Checking the sound. Can you hear me? Can you hear me well? So yeah, but I have to be like just really close. If I'm standing right here, the mic doesn't pick up. I have to be really close to for in the red. Right, well, but now it's good in the yellow. Okay, okay. So the mic is good. imamo jedan djelo obejna na evo što sve na mišlju. Evo sve. Ja sam budi sjeli. Shake, shake, shake. Zdaj da vidim, kako je to slišati na streamu, zdaj, ker me je šlo čisto do pječe. To bo zdaj. Treba si ključa da gledam, ne? Ja, što gledam. Pa je. Sam začel, pa malo krasno. Kaži, kakšno dobro, kaj s tabutom? Ama ne, to je bilo. Jedno koleno. Znao većinu da plus složeno, no, evo, pa smo rekli da malo boljši način, zdaj jaz malo tudi računam, da se sploh vidi. Boš ti šnemo te prezentacije, sploh točno se zdi, če boš videl. Kaj že, kakšna prezentacija sploh pošla? Evo je. Pa boš še pred tebe slikam. Ti sam se vpraša, kakšna pravo pravo. Ja, je. Kaj, smo te šli tako? Ampak je v strežniški sobi, pa ne printer. Io non avrei visto, non so.
Ja, bo do ciebie,
Streaming je že.
Riške tu.
Kako ćem to dati nekam? Recimo, da ne bo. Čak sam sekunda. To ugasnem. Ok, kadar sam.
Hello and good morning. Welcome in the morning session of speleobiology and geochemistry. So in the last few days we have some very nice explanations and lectures about karst, about physical environment, and today we are moving to life. Life is of course important, it's also shaped our karst. I would like to invite our keynote speaker, Slavko Polak, who is affiliated with Notranjska Museum Postojna, Slovenia. He is museum curator. He takes care of many biological collections. He's specialist for different animal groups, researchers, and lecturer. So Slavko will present us the studies on the cave animal DNA molecules can help us understand the formation and genesis of karst areas. Slavko, please.
Dear colleagues, dear organizers, thank you for inviting me to have the possibility to talk. Well, I will make a short frame about biology, about taxonomy, and about how this biology could be very helpful for Karstological studies. We all know that starting with the Karline, we start to give name to particular animal or plant animals, living creatures, and starting with the genus name and species name, and in basic zoological system could be classified as much as possible in natural frame. Unfortunately, in that early time, similar animals sometimes were put together because they were not already, at that time, it was not recognized. They are different, actually, very similar but different animal group. From this period, we can divide vertebrates to fish, to amphibians, to birds, to reptiles, and to mammals. Yeah? But because there are many so-called convergent evolution, sometimes we have similar animals which could be put together, but because of studies of biologists, we know now that they are actually far away phylogenetically in evolution, but they are simply similar. Like cases, this case of animals which are swimming, they are more or less similar, but are belongs each other to another group of animals. Much more known cases like Australia, we have that in Australia we don't have placental mammals, but we have these marsupials. But in Australia we can find wolves, martens, rabbits, moles even, and they are not related to European or other world creatures. Well, the new era started with Darwin theory about evolution of animals, and we start to make comparative anatomical comparative studies, and because of that they found that many things which look like they are actually not related. So now we know that Difference between fish is artificial group. Now we have sharks and bony fish, and they are very much apart, not very related. And then we have, well, I will say, especially reptiles are very terrible group. And years later, not very far away, scientists find out that birds are actually not a lone group, but actually just one branch of dinosaurs which survived this terrible situation 66 million years ago when big asteroid hit our planet and most of reptiles and dinosaurs were extinct, but only birds survived. And then came Corona time. We all faced with that, and you all faced with these sticks sampling for testing. Corona virus is actually COVID-19 special virus which made pandemic disease all over the world. We all faced with that, and you met with this. Yeah, this is COVID tester, but yeah, we have different testing. The first one is antigen testing. It's very familiar, very fast, but it's not very accurate. It's based on antigens, on antibody detection. Sometimes works, sometimes don't, and it's not very reliable. Yeah? So you face also that when you travel around the world, or maybe you want to participate some event, they existed that you have to have PCR test, not antigen test. So what is PCR test? Yeah? Well, this you may be faced with that last years because of this pandemic disease, but biologists and also medicine students, we use these molecular studies for maybe decades before. It is very useful. Nowadays, biologists apply modern phylogenetic approaches, studying these molecules of animals and plants to understand phylogeny. Phylogeny, that means, as I said on the very introduction, that we have to put animal and plant species into the natural system, in so-called tree of life. So related, phylogenetically related groups have to be together, and non-related have to be separated in this so-called hierarchic phylogenetic tree. Well, this method, you are probably not biologists, maybe few, not familiar with that, but method is actually seems very simple. We take a sample of tissue. It's one leg of small insect, of mosquito, it's enough, or one hair of us, and we isolate molecule of DNA. I will not explain what molecular DNA means. You know that, you're educated. And then we have a special machine, so-called, and we, in this machine, we reply a small piece. We put together also some primers to decide which particular part of DNA is interested for us, and we make a replication. After a few hours, we have this, PCR means polymerase. This is enzyme which open the molecular DNA. Then chain reaction. We start with chain reaction. We start to repeat one reaction to reply small particular part of DNA, which is interesting for us. And finally, after a few hours, we get 2 billion copies of tiny particle of DNA. Well, usually when you see some documentary films about genetics, you will see that this stuff and this observing with ultraviolet lamp and glasses, this is just testing if we get results. If we get this band, that means we get results. If there is a gap, that means we didn't reply this particular part of DNA. And very important is also control. If you have band there, then you have some infection or contamination. It's better to do everything in bucket. Otherwise, you have wrong results. Yeah? If we reply this sequence so many times in 2 billion copies, then we can easily use spectrophotometry, and we get this adenine, guanine cytosine and what I forgot, thymine results, and this is what we are looking. This is just combination of letters. After that, we have computer games, so mathematical programs, computer programs, and we compare these sequences. If we compare sequence of your sibling or your member of our own species, usually this sequence is the same. More related are more similar, less related species are much more different, because from time to time some changes appear because of mutations. Yeah? And nowadays biologists cannot publish any phylogenetic or deep taxonomical work without these phylogenetic, molecular phylogeny dendrograms, which are much more reliable than other methods. And sometimes these results are quite surprising. Well, we know that we have a group of birds we call the birds of prey. We have eagles, falcons, vultures, and so on. But due to this new method of molecular biology, we found out that falcons actually does not belong to birds of prey, but they are instead related to parrots. Yeah, nobody could imagine that, but genetics is 100% true, and nowadays we can find also morphological similarities between parrots and falcons. In these studies, it is very important also, as I said, geographical and geological barriers. Geographical barriers, now we divide our planet to Palearctic, African, Neotropical, Americans, and maybe you will see here in this next picture, we have porcupine, these are rodents, but we have two groups, one belongs to America and another one to Palearctic or Eurasia. Yeah. They look like, but they are not related. Also, geological and geographical barriers are very important. Here you have alpine plants, you have alpine butterfly species because of geography and also geological differences. Every this isolated part has its own species, but well, also this red line means ophiolitic zone, which is also very important geological barrier. But well, these alpine plants and alpine butterflies are not so old enough to study well geography or geology to help us, as I predict in our title of our speech. They are much more younger, mostly belong to this separation, these events during Pleistocene, which lasted 2.5 million years. You know that glacial periods were repeatedly coming and replacing with warmer climate, and that is very strong. That was very strong reason for this speciation. Well, now we came to the cave animals. Cave animals seems to be much more isolated because of they catch in cave environment. They have no gene flow between karstic areas, and this we expected years ago that this could be good group for deeper phylogenetical studies, and also a subterranean environment is relatively simple. There are not many environmental influences. Well, beetles, crustaceans, and so on. Yeah, our colleagues in Ljubljana University, they started with these phylogenetical studies mostly on aquatic animals many years ago, and they found very good and very well-known results, like these cave shrimp. Before there was a distribution of cave shrimp here in Dinaric karst, but they're also found in France, Spain, and also in Carpathians. And using this molecular approach, they found out that they are really, really similar, but they are not related. They are separated for millions and millions of years. So this is distribution of Proteus, which is also well-studied animal in Slovenia, and this is the only subterranean clam, Congeria, and you will find that we found that in Slovenia, well, the Proteus is actually have holodynamic distribution in all dynamic area, but there are different species here in Istria, in Croatia, in Dalmatia. So a similar appeared with this clam that genetics could be very, very useful for this. So maybe, yeah, do we know boundaries of, go back? Yeah, you will see that the dynamic karst boundaries are quite different from the outer to outer, depends which geographical or geological map you take. And here is my question, do we know boundaries of dynamic karst? You will say yes, and I will say no, we don't know. You will see later. Different authors make different maps. Of course, some are based on geography, another based on tectonics, another on geology. This is the most useful map after Roglic, that yellow or orange line, and another after Ivan Gams. You will see that it's much more precise. But both, they didn't put strict southern boundary of the Dinaric Pass. As I said, the role of geology is very important. Here we have ophiolitic, Dinaric ophiolitic zone, and also tectonics makes a very important role. Well, we started, this is case study, we started with one group of leptoderinic beetles. They are just part of beetles which we can be found in Europe. We started actually 19 years ago, and our study was finished last year, and it's still going on. Why so long time? You see I still have brown beard in that picture when we started. We have to collect fresh specimens for DNA isolation. We have good collections, but specimens in our collection are usually not well preserved. Well, DNA is decayed, and it's easier to go down there to Bosnia, to Montenegro, to Albania, Bulgaria, whatever, to collect fresh specimens preserved in pure alcohol, and it's very easy to extract DNA after that. So, leptoderini beetles are mostly subterranean. I said mostly, this is very important, because we have, in every group, we have some species which are not living in deep caves, but they are living in leaf litter, in forest litter, or deep soil, even superficial layers of karstic areas. And that is very important, since they are much less isolated. They can penetrate much easier to other areas. And distribution of these beetles are mostly on the northern part of southern Europe, and it's very obvious that on the northern part of Alps they are not. They are not, and we are believing that the reason, the main reason are glaciation during Pleistocene, which destroy all the taxa there, because we found evidence that there must be some connection. Yeah, so far, well, as I said on the introduction, we want to put species to genera to other families and so on, to make this hierarchy, so-called systematic taxonomy of the group. So far, these leptoderini were not, well, we put all this data of different groups on the map. It was a mess. Actually, it was very hard to explain real some features, that they are mixed. Yeah? And scientists still now, they change position of different groups from author to author quite seriously different. Yeah? And this is our results. After a long period of studying, we prepared this so-called planogram of European leptoderini. We sampled more than 400 samples from all over the world, and the results were shocking. Actually, when I got the first results, I said, shit, this genetics doesn't work. It's quite a mess. No, it's not working, because it's so strange. But when we put our data on the map, it was really clear. We have really nice geographical pattern of different groups. So here we have Cantabrian, Pyrenean, West, Spanish coast, then Tyrrhenian, Alpine. Interestingly here also, and so on. I will not go in detail. We found that before we talk about Carpathian fauna, but Carpathian fauna are divided in Romania to Apuseni Mountains. It's one fauna which is related to this from Pyrenees, and this southern Carpathian, which is quite Balkan one. Yeah? We found that the oldest groups are evolved. The group probably evolved here in the Balkans, since because this Adelopsiella bosnica is distributed here in Bosnia and Montenegro, and the next very old group appeared here in Balkan Planina in Bulgaria. So this is the oldest separation. Actually, we believe evolution of this group started probably here. We also dated, and you will see here that we say that this evolution of this group started in early tertiary, because the out groups of the Leptorini live even in America, in California. So it means that ancestors of group have to disperse to American continent. But it happened during early Eocene climatic changes. Ancestors, well, this is the picture. We calculated separation time when these groups are separated, and we found that the group of subterranean beetles are much older than we expected before. How do we know? Yeah, Italian authors Caccone and Sbordoni many years ago put one theory that, well, beetles from Sardinia is the same genus as these beetles, Speleonomus beetles in Pyrenees. And we know it's well documented that this Sardinia and Corsica separated from this Spanish, Ligurian plate about 20 million to 30 million years ago. And that origin of these beetles helped to travel to these islands. And so this is good, we call this genetical clock. We calculate how many mutations could happen in this period. And that is useful also for explanation for other groups. Yeah. Well, I will not go in details since maybe I will too long. We changed these groups of Leptorini beetles very much. Excuse me. Yeah, you will see that, as I said, here in Southern Carpathian, beetles are definitely not Carpathian. It's much more related to this group, or maybe Rhodopian group, similar this one, or Carpathian group. And Rhodopian group is also here around the southern Balkan, but well, on the really Rhodopian mountains, there are no Leptorini beetles, since we expect, because group is very old, the karstic cover of Rhodopian mountains already dissolved where this group started. Yeah, also one very nice group is Alpine group, which is just yesterday Theo Delic sent me new results, and now we say that these triangles are actually right positioned. So this study is going on. These alpine beetles have to be limited to alpine region of Slovenia, but you see this red dot, this is Aphaobius, which penetrate down there to Dinaric range. We can explain that because Aphaobius not live only in caves, but also in leaf litter, and during Pleistocene time there was much more humid environment. So this genus, only one species, one group penetrated deeper to the Dinaric. Otherwise, other species are limited to alpine region. Yeah? And Dinaric, we are mostly focused on Dinaric group. Here we have what we found. We found that there is no one Dinaric fauna. Instead, we have two Dinaric fauna. Northern Dinaric, this is, we sampled almost all genera. And next one is South Dinaric. And this southern Dinaric fauna is actually made of two groups. One is really Dinaric, and one is much more so-called maybe Hellenic or Aegean origin. So that is this open new possibility, new questions about simple Dinaric range organisms. Yeah. And if we isolate these others which are not dinaric, if we put only dinaric genera together, we put that data on the map, and we can get really sharp border of boundaries of dinaric karst. Yeah? Here we can make a determination of southern part. It is strictly near the Skadar Lake, then this Prokleti Albanian mountains, small part of Kosovo around the city of Peć, then most of Montenegro without this most eastern part. In Serbia, only small part could be declared to dinaric, and then we have quite very nice border of dinaric karst. Here you have some question marks. You have to have good knowledge on taxonomy of these beetles to extract some questionable data because of mislabeling or also misdetermination. Yeah? Well, if we compare, I said, now we can make determination of this southern border of dinaric mountain. Yeah? Yeah, surprisingly enough, even on the northern boundary of dinaric karst, we have to, well, we found that there is some contradiction. These are mapped after GAMS 2001, GAMS 2003, and Busser 2010. We always talk about dinaric karst, alpine karst, and in the middle we talk about so-called predinaric or isolated karst. But, yeah, if we take this in consideration beetles, we found that these beetle, Celtmonocharis, live around Dobjale karst and around the canyon of Sava River. So it's actually strictly dinaric animal, and there is no other alpine animal penetrating this area. So we can put another, well, we can put strict border of northern boundary here, and then near Ljubljana, Ljubljansko-Barje, then Idrica Valley, and the dinaric range stopped on the Banšice plateau. Not only beetles, also other groups of animals are similar, are very interesting, but we found that there are quite significant difference between terrestrial subterranean animals and aquatic terrestrial animals. And our colleagues in University in Ljubljana make many results. So many groups are still not studied. There are many aquatic and terrestrial groups. So in the future, this will be very important issue, and we already discussed about this orography and these reasons for these distributions with geologists like Bojan Otaničar, with geographists like Andrej Mihelc. I actually took Andrej Mihelc twice with me to Bosnia and Montenegro to explain me what happened, and he said, you biologists, you always accept the world as static, but geologically and geologically, these mountains were floating, digging, you know, this karstology is a never-ending process. Also, Professor Platzer was involved in our studies, and well, we are biologists, we don't have this knowledge, and we are inviting you for debate, and even to make some common contribution in future, to put our data together and to try to explain orography of these areas. Thank you for your attention.
Slavo, thank you very much for this nice talk. Is there any questions?
Of course, there are. Actually, every study opens more questions than answers.
Slavo, thank you. I was wondering on your map where you were.
Microphone.
No, I was wondering on your map where you were delineating northern and southern Dinaric. Can you tell us more about this zone that was having the two species, the two
Crossover, yeah. Well, it's surprisingly that in this between northern and southern dinaric fauna, we have quite strict fauna line. There is a bit of overlapping, but this overlapping is a reason for these species which are not living in caves. The species which are living in littoral soil, and there they have much more ability to cross. Otherwise, if I exclude this line will be very strict, and it is approximately line Sarajevo split. Tell me, what is that? We have no idea. Geomorphologists and also tectonics always divide external dinarics, but this is in this dinaric direction. What is in this direction, we don't have idea.
Thank you very much, Lauko. We continue with our presentations. I would like to invite Anja Paladancic to present her study. She is affiliated with two institutions, Natural History Museum from Vienna, Austria, and Biotechnical Faculty, University of Ljubljana. She is expert in zoological systematics, evolutionary research, and phylogenetic relationships. Please.
Hi. So I hope you were paying good attention to the first talk, because basically, maybe then I don't even need to do mine. Yeah, we got this project on disperser, this time of aquatic organisms on karst landscapes. And yeah, I will start. Of course, I don't need to tell this audience about tracing tests. Yeah, the tracers are where the water goes, but also where like the tracers are the same as the pollution goes. Yeah, and that's why it's interesting for us biologists. The thing is that now in karst in general, but also in binary karst, there are a lot of endemic and that's why also endangered animals, and they are very vulnerable to pollution. So when we have a pollution of one part of one water source, it can easily spread to the karst aquifer. And the second thing is that, for example, there are some fishes that would live only in one karst polyet in one specific place, and you can imagine what happens if that is destroyed. So, in many studies, previous studies, there was a question for organisms. Are organisms like bounded by karst? Are they is karst actually an isolating factor of organismal dispersal? In the previous talk, we heard yes, because they would be bound to caves. But we found in literature different answers, and the most important actually is the spatial component. So yes, partially it is an isolating factor. It depends on the scale that we are looking at. So the aim of our project was to test that. Yeah. So does underground migration through the water sources influences the dispersal of karst organisms, or does it influence their dispersal areas? And what was also interesting for us was, could we use this population structure, so relationships between the organisms, how related they are, to suggest also some input on the water connections. And thus we took 3 groups of organisms, because organisms are very different. So we wanted to choose different organisms in matter of size, in matter of movement. So we choose fish and we choose proteus, which are quite big organisms. They are vertebrates and they can actively swim. And we also choose some smaller, like Azelus aquaticus, which is an isopod crustacean, invertebrate, and it has possibly it is passively carried by the water. So these are our beautiful organisms, and in addition to the size and to their we also wanted to see the different biology of the animals. Because karst, of course, is mostly known by cave organisms, but there are also a lot of other organisms living in karst area which are not bound to caves. For example, we have these fish, like minnow-like fishes. Yeah, they are not related to each other, even though they look a bit similar. And some are like completely accidental occupants of caves. They would be carried in caves by the water. But some, they are at least partially bound to the caves. So in the lower, like in the middle part and the southern part of the dinaric karst, there is a group of fishes which are not related to each other, but they have a similar way of life. So they would be bound when the water is low, so when the surface water dries out, they would actively survive in caves. They do not look like cave animals, and they spawn outside of the cave, so they still need this dewary lifestyle, partially caves, partially outside. And then we have, of course, typical cave animal, completely cave animal, and in the Zelus aquaticus, there are different forms. So this was also interesting. I have to disappoint you at this point. I don't have all the data yet, so please don't expect me to tell you all the results. We focused on two different smaller study areas in the Dinari karst, mostly also because we have amazing tracing tests at these areas, especially in the Dinari karst of Slovenia. And the second part is between Livno and Sin, and there are also tracing tests done there. So yeah, mostly for the fish, yeah, but also for other animals, we suspect, let's say we had three hypotheses. First was that this population structure that we see reflects current underground water connections. The second was that that we see some part of past hydrological conditions that was left here, and we can see it in our population structure, or it's because it is fish, and even though that they are not commercial fish, these minnows, they can still be translocated. So it's also possible that population structure that we see, or some strange patterns that we see, are a consequence of anthropogenic translocation. Yeah, so what we chose was again genetics. As we heard previously, genetics doesn't lie. But yes, we can still be wrong, that I have to still say. So we just wanted to see, based on genetics, based on which organisms are related, what does this population structure tell us, and which part of the hydrology maybe it reflects. And as we heard, yeah, the boundary of the Dinari karst is set, but maybe not, it changes, yeah, or it's not stable. But here are some of our first results. So this is our minnow, and remember, it's not a cave animal. This is only accidental cave occupant. But we see, yeah, that it has some clay, some different colors. You can just follow the colors to see different clays. Some of the clays are spread really far, like the green one. They are spread far out, long into the Dinari karst. And then we have, and these are the newest findings of the Proteus, of the genetics, and then we have these clays of Proteus. How are they spread? And we see some small overlaps, but I would not say we see a general pattern. And then we have also from previous studies, it's crayfish, which is also not subterranean, so it's not a cave animal. And I would say that in comparison, maybe that's a better comparison to what we see, right, in sense of clays. Again, just follow the colors and you will see how they are spread. And as I said, we wanted to take a closer look in this area in Slovenia, and it helped us tremendously that in 2020 all the tracer tests were digitized. So it was possible to do this kind of maps, yeah, and to compare them then to our population structure. We sampled everywhere where we could find this fish, and that's also a plus of small study area, because we really sampled everywhere where we could find the Proteus, minnows, and what we found was that there is some sense some patterns in these connections that we see, right? So this, if you again follow the colors, the same color represented means that it's the same, let's say, genetical sequence that we saw previously in this clade, right? And what we also noticed is that out of this karst area, it's not depicted here, but the genetic signature is quite different. So we see, in general, more connectivity between populations of the Dinari karst than outside, let's say, also in the alpine karst. Yeah. So, and then in comparison, yeah, with Proteus, maybe we can see these two clades, right? So here we see a lot of green going on, and here is something completely different. And maybe we could see the same here and here, right? So there are two clades, but we see that it's completely different. So the boundaries are different. We don't have this special clade here. We see, for example, that between these sides, where we know there are nice connections to based on mitochondrial DNA and not based on genomic quicker. So I can put maybe a timeline on this point and say, they were connected previously, but they are not connected anymore at this point in time. So in the second part, unfortunately, we don't have such good data because we only have museum samples. I think from if the reports from the field people are correct, I think there are no more fishes to find in Sin, or they are very rare, so they are possibly extinct at this locality. But in our nice collection, in the fish collection of Natural History Museum in Vienna, have a lot of fish, like hundreds, thousands of fish, and some of them are from Livno and some of them are from Sin. So we use those to do our genetic research, which is of course a bit more challenging, but it works. And here are our results. So these are two different, two other species, and in the populations of the two from Livno and from Sin, we basically see no differences between them. So we can say that they are possibly closely related. But as I said, this is very basic data because it's very hard to work with old DNA, let's say. And we also had an idea that in these middle points that are between our sites, we would collect water, extract DNA from the water, and check if there is DNA of our organisms in the connecting water from the surface sites. But to be honest, I don't know if we will have time to do that because the project is soon. So now we are at this point where I said most of our current data, which we also published, was based on mitochondrial DNA, which is more basic type of analysis. And now, at this point, our student is working on genomic data, so to compare the DNA, so genetic signature of the whole genomes between the populations. And the most important for us would be, of course, to do the timeline of these splits. So that would be everything from my side. Thank you for listening. And as I would just repeat the last sentence of the previous speaker, of Slavko. We would be, of course, very happy to be contacted because geology, hydrology confuses us. Thank you.
Thank you very much. Do we have a question, perhaps?
Thank you. I listened to it with great interest. I'm from New Zealand, and we have used the genetics of fish to tell us about the evolution of our river basins.
Yes.
And here you are in a very tectonic situation, just as we are. And the stories that you've been telling us about the distribution of these species will also tell you something about the development of the hydrological systems, which was connected to which and when did they separate. Can you tell us anything about that, please?
Well, I can only tell an anecdote. So we contacted, of course, some hydrologists and geologists, and we asked them, we said, oh, we are having this project. It's very interesting. Could you help us? And they said, oh, no, you can help us. Because it's apparently, maybe I will explain this wrongly. I'm so sorry. Like I said, I'm a biologist. But it appears that Dinari karst is very quick in dissolving, in changing, right? So it's also for the geohydrologist, it's a very hard process. It's almost impossible to pinpoint times. But also, to be honest, we also need these times to understand our structure. So I'm not really sure. And what I also, maybe I didn't emphasize enough, also what we saw in the previous talk, it's one species. We have now a few species. It's very dependent on the biology of the species. That's why we have to take care of this always in the background, right? Because if they are terrestrial, if they are aquatic, if they can move, if they can just sit, it will tell us a different story. So, in this part, the biology of the species and how much influence it has on the migration, it's the hardest part, because it basically, I suspect that every dispersion pattern in these groups of species will be different. Yeah. So, but I really hope that we will find at least some points. Like I said, we in the genomic era, they promise that a person can pinpoint this split between populations in the timeline. So hopefully this will help us also to understand a bit what happened and when.
Thank you very much. I would like to invite Qiu Fang He from Institute of Karst Geology, Southwest University, Chongqing, China, to present her study. She is actively involved in karst ecosystem and particularly in carbon cycle. Please.
Ladies and gentlemen, very happy to give to introduce our work in Lijiang River. We are focused on the organic carbon sink in this area. At the past, we are working about the inorganic carbon sink. As we know, in the karst area, the carbon sink are come from the carbonate weathering, and they will become inorganic carbon and stored in the rivers and the reservoirs. So they will become inorganic carbon. But nowadays we found that the inorganic carbon will encourage the aquatic plant growth, and then they will also store in the as a kind of organic carbon in the plant and in the dissolved organic carbon and particle organic carbon in the rivers. So here we want to know. What the organic carbon forms in these aquatic karst systems. These are the simple modelings in karst aquatic systems, and the dissolved inorganic carbon will formed by the photosynthesis about the aquatic plants, and they will become 3 parts of the dissolved organic carbon. The first one is from aquatic plant, and the second will be microbial formed, and the third one will be comes from the soil, because of the soil erosion in China is very serious, especially in the summer, because they are infected by the Asian monsoon. There are the precipitations and the rain and is very serious in the summer, and a lot of soil will rush into the river. So we want to know in the Karst rivers, these 3 parts of organic carbon, what's about the proportions that will be useful to calculate the organic carbon sink in this area. But now we do not know about this. And another is, organic carbon from the animals and the plants, they will be degraded by the animals. They will reconstitute into the microbial organic carbons. Does this happen, and what's the mechanism here? So we take samples in Lijiang River from the upstream to downstream, and we connected all the samples to test the hydrochemistry parameters, and also used the stable isotopes to find out the organic carbon sources. And also we do the bacterial diversity to find out what happened to the bacteria and organic carbons. Our question is to find out the composition of carbon sink in this river. How many comes from the aquatic plants, how many come from the microbes, and how many from the soil. And the other is the cycle mechanism of microbial sourced organic carbon. And these are the methods we have used. And in Lijiang River, we can see the dissolved organic concentration will increase from upstream to downstream. And we know the substance in the Karst River will come from the surface and the subsurface. So we tested the concentration of dissolved organic carbon in groundwater. We found is relatively low, just as same as the upstream. So the dissolved organic carbon will come from the surface part. But the concentration increase is because of the autochthonous or heterogeneous. The heterogeneous means come from the soil. The autochthonous is come from the aquatic plant or microbial. So we used the colored dissolved organic matter spectrum to help us to find out the component of the organic carbon. We identified these 3 components, and the first one is aquatic plant, heterogeneous, and microbial. And we used the fluorescence intensify to represent the concentration or something or participation of these 3 parts. And we used a linear regression to calculate the concentrations. Finally, we got the concentrations from upstream to downstream about these 3 parts of organic carbons and their seasonal changes. Also, we used the isotope mix model to find out the proportion of these 3 parts of organic carbon. From this, we can find the proportion of microbial source organic carbon will increase, and then the 2 parts are decreased, especially in the middle, in upstream to midstream, 3 parts are increased, but at the midstream to downstream, the input of soil organic carbon is almost 0, because in the middle to downstream, the water is relatively very clean and almost no soil or input. So we think the spatial increase of organic carbon resulted from microbe and aquatic organic carbon, and what are the critical factors impact this organic carbon recharge. Here we can find that as a very similar temporal trend of total organic carbon, soil carbon, and discharge and precipitations, especially significant positive correlations were found between the turbidity and the soil organic carbon content that we can find that the turbidity might be the most important reason that will influence the soil carbons, and the turbidity increase mostly comes from the precipitations and storm events in summer. We can see in summer there are very, very high soil carbons, and the aquatic plant organic carbon are found the significant positive correlations with chlorella A, and which is the important reason that the phototrophic synthesis will induced to the growth, plant growth, and aquatic carbon increase, plant growth, and so we think that will be the temperature and illuminance which influenced the aquatic plant organic carbon, because in the spring and fall was we found the highest plant organic carbon, and in the winter the temperature is relatively low, the plant growth were limited, and in the summer, because of the turbidity increase, the illuminance in the river will decrease. So these are the two reasons influence these two parts. But how about the microbial carbon? We can find that they are both of the plant organic carbon and the microbial organic carbon are increased with the dissolved inorganic carbon. In the past research, we found that the high concentration of DIC will encourage the plant growth. So this soil source organic carbon will increase, and then the microbes go up together with the plant carbon. And I think the microbial organic carbon increased from upper to downstream very, very steadily. So what's the mechanism? We found we tried to find some information from microbes, and we found some microbes lineage are correlated with the soil carbon, and some others are correlated with the microbial soil carbons. And these two carbons comes from different seasons. The soil related bacteria was found very high relative abundance in the rainy season, and the microbial chlorinated bacteria were found very high concentrations in the season before. Oh, sorry. And most of these bacteria are heterogeneous bacteria. So we think these are bacteria was reconstituted the plant organic carbon, but not from the organic carbon from the dissolved inorganic carbon. We use the peak crust to predict the metabolism pathways in these rivers, and we found some of bacteria. All of this. We found this all of this organic carbon synthesis pathways are found a very high positive concentration with this sporage stasis and novel synfil bariums. These are two very important bacteria species will influence the organic carbon. And then the heterotrophic bacteria degrading the deniable dissolved organic carbon and form the microbial organic carbon. We used a level calculation experiment to find out what happened in the organic carbons. We found three kind of organic carbon were changed in the calculation period. There are significant decrease in about the plant organic carbon and increase in about the microbial carbon and soil carbon. So these are our conclusions that the increase in Lijiang River comes from autocolonious organic carbon, heterotrophic bacteria contributions to the microbial increase. And the DIC was encouraged both plant and microbial organic carbon. Thank you.
Thank you very much. Do we have perhaps a question? Thank you. We are very much on time. I would like to invite Marcela Hernandez Garcia. She will have an invited lecture. She's coming from School of Biological Sciences, University of East Anglia, UK. She's expert in metagenomics, biogeochemical cycles, microbial ecology, and biochemistry. Please.
Yeah, hi everyone. I have my timer here, so I hope I'm on time. My name is Marcela, and as Gianni said, I work at the UEA. I thank all the organizers, especially Gianni, for allowing me to be here today. He said, I have to give you a lecture about metagenomics. So I thought, oh my gosh, because I'm not even, first of all, I'm not a geologist. And secondly, I don't work with karsts, nor with caves yet. But I want to put you all out of your comfort zone and start to understand more about sequencing, metagenomics, and yeah. Basically, I work with label trace gases, and I combine that with metagenomics, and I analyze the active microbial communities from extreme environments, such as volcanoes. All right, so the microbial tree of life. So the microbial tree of life represent one of the most interesting concepts in genomics, right? And as you can see here, this microbial tree of life is separated between three main domains, yeah? And this diagram was taken from a book from 2009. So here we have the three main domains, bacteria, archaea, and eukarya. We are here, fungi is in eukarya, and then archaea, there are several microorganisms that they work mostly with methanogens and ammonia oxidizers, for example. And bacteria, with bacteria you have everything, right? But as I said, that was in 2009. So what has happened since 2009 to now? We have many, many phyla that have been identified, and the domain is a little bit different, and they all come, as I hear, they all come from a main universal ancestor. Now, why I'm showing to you this graph today is because nowadays, so that was published by Laura Hack in 2016, and she identified 92 bacterial phyla, 26 archaea, and 5 new branches of eukaryota. So, but the most important thing here, and why this is linked to metagenomics, is that all these red dots that you see here everywhere, those were lineages that lack an isolated representative. So she identified that by using metagenomics, right? And but those dots don't have a genome available, accessible for us to work. So this is why it's so important to combine wet lab, which is like enrichment and isolation, together with metagenomics, right? So now, metagenomics. So why do I combine metagenomics? I already explained to you that. And now I want to explain to you how we analyze metagenomics in very friendly words so that everyone will understand. So first of all, think about this cotton thread I have in the bottom. Is full of different colors. So this is like DNA, right? From any material you are working with, either human or soil, tree, atmosphere, even water, etc., gut microbiome, etc. And then every separate cotton thread of every color, it represents a genome, right? So then that's, I think, is an easier way for you to understand metagenomics. So we have a sample, any material that you're working with, we extract the DNA, and now it depends on the sequencing machine, then we will use different ways of extracting the DNA. Yeah, if you are doing PacBio nanopore, then we won't be using mechanical disruption and not to shred the DNA. But if we are doing Illumina, then it's fine, right? So we will extract the DNA sequence, and these are all these fancy machines that people think is very expensive, but now it's not too expensive. In 2000, all right, no, I will explain that later. So, right, so we extract these sequences here, which are little pieces of DNA. And then computational programs, we use high-performance computing cluster, and then with assembly, it will stitch pieces of DNA. So it will use a different program, and it will start identifying nucleotides, and then it will stitch together pieces, for example, the green ones. So it will stitch it here, and then another here, and so on. And then the same with the blue, and the same with the red. And then during the assembly, what happened, it will group those pieces of DNA. So it will match them, and then it will start building or retrieving what we call metagenome assembled genome, which is this independent cotton thread that I showed you in the bottom here. So then happy days, because we have genomes that were retrieved from my material. So we can now assign function, for example, and answer our scientific questions, right? So then what we do functional annotation. And when we do functional annotation, then the program will identify open reading frames. So it will translate the DNA to protein, and then it will identify a start codon and a stop codon, and then it will annotate each of these genomes. And then with that information, then we can do metabolic reconstruction, and we can then analyze our metagenome, right? So super clear, basic words, happy days. If you don't know, if it's confusing, then we can collaborate, right? So it's fun, it's easy. Right, so, but why, how am I here, actually? So in my work, you can see here, this is a picture from Yaima volcano. I work with volcanoes, and mostly with volcanoes from Hawaii and volcanoes from Chile. And I analyze the role of the microbes supporting soil formation. So soil formation, you are all geologists, so I'm here. I'm in my not in my comfort zone right now. So the soil formation is a combined process of physical, chemical, and biological factors, right? And then you know all of that, and we can do a whole conference on that. But my contribution as a soil microbiologist, environmental microbiologist, is to look at the contribution of microbes, specifically how microbes can oxidize trace gases from those extreme environments. And I combined everything, lab with metagenomics. And so I look specifically at trace gases. So we have methane, we have carbon monoxide, we have CO2. And for that particular project, I work with the carbon monoxide dehydrogenases, which is the enzyme which oxidizes carbon monoxide. So it will oxidize carbon monoxide to CO2 and then electron and protons. And this is going to be used as energy for the electron transport chain, for ATP production, and to the Calvin cycling for CO2 fixation, right? And then, right? Yes. And then the carbon monoxide dehydrogenases, we work with this specific group of genes, the COX genes. And then, yeah, we have different type of carbox carbon monoxide oxidizer, the carboxydotrophs and carboxydovores. So they are chemolithoautotrophs. So the carboxydotrophs and the carboxydovores, they require an additional source of energy. And why carbon monoxide is important? Because it's toxic. We all know that. 40% of carbon monoxide is produced in the environment. And actually microbes, soil microbes, are able to consume about 250 to 300 million tons of carbon monoxide per year. So it is important. And so quickly what I do, by using metagenomics, I identify all of these max, so from different sides of different volcanoes, so either Kilauea volcano, Llama, Calbuco volcano, etc. And then I analyze the operon of these genes which encode the carbon monoxide dehydrogenases, so potentially oxidizers of carbon monoxide. Now, why am I here again? So that, what I told you before, was all very nice, but it's an open environment, right? So even though it's this extreme environment, we have the effect of light. A colleague of mine, Colin Murrell, here, and myself organized a workshop in Norwich. And because I've seen pictures of you with your friends, I thought I'm also going to put pictures of me and my friends. And during that workshop, we invited Sarah, Andrea, and Yanni to participate in the workshop. And that was in February. So over the 4 months, we set up a collaboration, and we started to somehow support Sarah's thesis. And then I was invited to be here to teach you about metagenomics. But then I thought, but why don't we do metagenomics in karst environment? And then I tried to give you something more than just a lecture on metagenomics, but how we use it for caves. And so the title now is using metagenomics to study cave environments. And metagenomics now is 10 times cheaper than 7 years ago. So it's something that we can do. Not easily, but we can do it. And this is a work, mostly the sampling was done by Andrea and her group and Yanni. And it's a microbial activity in alluvial sediment in the Reka River. Now it comes Scotian caves. I've been like 5 hours trying to pronounce that name. Right. So for the sampling campaign, what they did, so the Scotian cave sampling site. So we have 7 different sections of the cave. This one, I will not even dare pronouncing this word because Scotian took me so long. So yeah, MRTVO site. Okay, so that side, and then this one, that, blah, blah. So sediments were sampled along the Reka River, which sinks the Scotian caves, and then this site 7 is from the soil surface, okay? So we identified 32 metagenome assembled genomes, which contains genes encoding the carbon monoxide dehydrogenases. And this was very, very interesting. I was really happy. So I will say we have approximately 50 or even more metagenome assembled genomes. But to answer our scientific question, I was just focusing in the carbon monoxide for this talk and for my research. So we identified 32 max. Now, with metagenomics, we can also see who is there. So here in the site 1, site 2, and site 7, site 5, which were like sediments in the river under the cave, we have high abundance of proteobacteria. So all of this is proteobacteria, here proteobacteria, and then all of that 58% is proteobacteria. And then we have a little bit of actinobacteria here, and then we have nitrospira here. And then actinobacteria is just like 25%. But now when we look at the soil surface, then it's mostly actinobacteria now, and proteobacteria is very little abundance. So I have 3 minutes, yeah. So I use, first of all, metagenomics just to have a broad classification of what we have. And then when I look annotation of these metagenome assembled genomes from the caves, then we have several marks. So all of the blue dots are the marks that we have retrieved from the caves. The green are type strains. So we do this annotation with type strains and housekeeping genes and all another thing for another lecture. But if you see the blue here, we have marks that align to this species of Limocorda, which is an isolate from lake sediment. So we were not too lost. That was good. And then from this is sample 7, which is the soil surface sample. and aligned with Conexibacter, which was isolated from soil, so we were good again. And so on and so forth. So then all of these marks in the bottom, they are from water, and they match with type strain from either groundwater, sewage sludge, or oligotrophic sediments. So, but most importantly, we have all of these marks on the top. They don't have any, they don't align with any type strain. So those are potential, what we call then in microbiology, we will call it potential candidatus of something. So that opens a window to go back to the lab and try to isolate, because remember the slide number 2, I showed you full of red dots without a genome being isolated. Well, then we know how we can isolate them, because we can go to the genome and we can identify pathways and then find a strategy to make an enrichment, right? And then increase the phylogeny tree of life. Right, I don't have conclusions now, but I have projections, because I mean, this project started 3 months ago, and I wanted to combine volcano, carbon monoxide with caves and new collaborations. So future projections, which microbes play important role in cave environments? Do they actually oxidize trace gases like methane, carbon monoxide, CO2? Which type of microbes live there? Do they do something else? Antimicrobial resistance, heavy metals, etc. Anything, any scientific question you have, we can answer with the help of metagenomics. And if it's trace gases, we can answer even combining that with labeling and doing stable isotope probing. So yeah, many scientific questions open here for discussions. And then thank you very much to Andrea and Yanni for inviting me to their project and to help a little bit, and to Dr. Chamik Roy, a postdoc working in my group, and who helped with the metagenomic analysis, because in 3 months, it's just almost full time of doing metagenomics. Yeah, thank you very much.
Thank you very much, Marcela. Do we have maybe a question?
All clear, excellent.
Everything is clear. Thank you very much. We continue. Please, Jo de Vele. He's coming from Department of Biological, Geological and Environmental Sciences, University of Bologna, Italy. He is well known in karst community. He's expert in many fields, and he will present the work Sulfuric Acid Caves of the World, a review.
D'Angeli. Doesn't start. Okay, I'm not Ilenia, as you see, but I'm presenting this on behalf of Ilenia. She did a PhD thesis on sulfuric acid caves in southern Italy. To do this, we had to go through the literature of sulfuric acid caves in the world, and then we came up with a review paper we submitted a couple of weeks ago, together with Filippo Dra, Arthur Palmer, and Lucas Plan. So I'm presenting more or less this. It's amazing to think that the first time someone published something about the role of sulfidic waters in the formation of a cave was in 1801, the start of the 19th century, Soquet in the south of France, in Aix-les-Bains. And this is Filippo Dra sampling the thermal sulfidic stinking water. He always jumps in that water when he sees it. Martel, in more than a century after that, talked about the same cave in a hydrogeological meeting in Paris. So also Martel knew that sulfidic waters could be important in the formation of caves. Krauschöhle, the end of the 19th century, was discovered by Krause. Krause himself and Hauer already linked the genesis of this cave to the presence of a sulfidic spring 80 meters below the cave, even though later on many researchers thought that the cave was formed by the dissolution of a large gypsum lens in the limestone succession. Later on, we understood that this is a really very interesting sulfuric acid cave in Austria. In Triponzo, in the central part of Italy, the digging of a gallery for a spa intercepted an active, small little cave, and that was described by Principi. This was the first real, very nice geomorphological description of a sulfuric acid cave in the world. This paper was published in Grotte d'Italia. He talks about gypsum, about notches, about corrosion table, and all this kind of stuff. Unfortunately, this cave cannot be accessed anymore. The first real description was Morehouse in the United States, Lower Crevice Cave. It's a sulfuric acid cave related to the oxidation of metal sulfides, but this was the first time that the whole chemistry was more or less described in a very nice way. National Speleological Society Bulletin. This is the year I was born, so 1968. This oxidation of sulfides in this carbonate sequence is important for some caves. Normally it's very local phenomena, very small caves, like the one we have seen before, Ibuciere. But in Brazil, for example, the largest cave system in the world, in the southern part of the world, is made mostly by the oxidation of sulfides in the carbonate sequence. So Toca da Boa Vista and Toca da Barriguda. Egemeyer, Steve, was the first who really described an active sulfuric acid cave in America, the Upper Cane and Lower Cane caves. Still a lot of people go there to study. Egemeyer did this in '73 in his PhD work, but also a couple of years before, in 1971, he was the first to think and to talk about Carlsbad caverns being probably a sulfuric acid cave. After that, in 1986, Lechuguilla was discovered, and this boosted a lot of cave scientists to go and work in sulfuric acid caves. There was a lot of debate on this. Where does the gypsum come from? How does it work? There have been a lot of theories converging into one because of the work of Kirkland, first of all in '82, and then especially Carroll Hill and Polyak, and even the Palmers. And now this is more or less the place where the whole theory was devised. The Guads, so many caves in the Guads are sulfuric acid caves over a very long time span, by the way. These have been dated by allanite dating. The same year of the ledge discovery in Romania, the Movile cave was discovered. It's a cave which is isolated more or less from the outside world, so there's a lot of places with low oxygen content in the air. And the first people going in there, they found a lot of microbial mats and material, and especially these strange animals that were adapted to this chemolithio-autotrophic environment and ate these sulfur bacteria. These were described in Science, and this is when the people started to understand that active sulfuric acid caves are really important places for the study of microbes, especially, but also cave animals that are adapted to this very extreme, sometimes very acidic environment. Biovermiculations, biological mats, white filaments in the water, everything. Most of the studies today in sulfuric acid caves are made by microbiologists. Sarbu, which was one of the cave scientists studying Movile, came to Italy, to Frasassi cave. This boosted the study and the search for sulfuric acid caves in Italy, especially by Sandro Galdensi. And in a short lapse of time, the whole Apennine chain was filled with dots of places where we found gypsum in the caves, and we knew that these were ancient sulfuric acid caves. I can call Italy the land of SAS caves, of sulfuric acid caves, because there's now more than 26 places in Italy where we found caves that surely were made by sulfuric acid. Now, I'm not going to waste a lot of time about the processes involved, but the H2S is mostly responsible for the formation of sulfuric acid in a continuous way over long time periods. The H2S comes from below, can be produced by volcanic magmatic activities in some places, but most of it is due to the bacterial and thermal sulfate reduction at depth of gypsum in presence of hydrocarbons. So most of the places in Italy, for example, are related to this process of H2S forming, rising up into the caves in the oxidized environment. This oxidizes, makes sulfuric acid, and carves the caves. There's a lot of reactions going on. You see this in this simple diagram. It's not really simple, but everything goes on, especially in the oxidized environment, so close to the water table, above the water table, and especially in the condensation water films on the roofs and the walls. This reacts with the limestone and the host rock, which can also be not pure limestone, creating a lot of byproducts which are very, make it very recognizable in caves. How can we recognize them? If we find these large gypsum deposits, that is a good indicator, but that is mostly not enough. Normally we need a sort of combination of indicators. Mostly it's minerals, secondary minerals, the geomorphology of the caves. If we find the water, it's much easier. If we find sulfidic water still in the cave, that's okay. And then there's some geochemical indicators of sulfuric acid caves, especially when they are fossil and there's no more water in there. One of the main things is, for example, the sulfur isotope imprint. Normally they have very negative values, not all the times, but many times. If it's bacterial sulfur reduction making the H2S, these sulfur isotopes go to very low values. You see a lot of places in the world where these sulfur isotopes have been done, and most of these values are below 0 and even up to -35. So this is one of the things that makes them recognized pretty well. Another thing is the secondary minerals. Sulfuric acid reacts and makes sulfates, especially gypsum, which is normally present in these caves. Gypsum is very soluble, so in old caves it can be washed away. But then you can find other sulfates like jarosite and alunite, the alunite group minerals. These are very important because they can be dated using the potassium-argon dating method. And these have allowed Polyak to do this in the Guats. We can go back for millions of years. And a very nice thing about these datings is that we are really dating the exact timing of speleogenesis. That's the moment in which the caves were forming. That's one of the few methods we have. There's dolomite dating as well. And then, of course, there's the morphologies. Most of the time, the waters coming into sulfuric acid caves can be thermal. So there's a lot of morphologies typical of hydrothermal caves, cupola and all this kind of stuff. There's a slow water movement, so it brings to a spongework morphology of the caves. But the most diagnostic morphology is the one on the right, the bottom. These are the replacement pockets, so the gypsum that is forming in small pockets that then falls away. And these are morphologies that are unique to sulfuric acid caves. If we find these and we find the gypsum, we can be sure this was a sulfuric acid cave. Now we did this on a literature review, so we looked in the world where do we have these caves. It has boosted since Lechuguilla. So many places have been found in the world where caves that were previously thought of being epigenic, normal caves, were found to be sulfuric acid caves. There's a lot of North America. There's something in the south of America. There's a lot of it in Europe and Asia. There's something in Africa. There's even lately one cave in Papua New Guinea done by French people. And in these days itself, I found 2 new places where we have sulfuric acid caves that were not published before. So this is really growing fast. Some interesting things, Albania and Greece. The last 5 years, many cave systems have been discovered with sulfidic water inside. Lengarica and Sarandoporo is close to the Greek-Albanian border. And Lengarica is already 7 kilometers of caves that have been discovered over there, still going. Holtas is in the center of Albania. We are at about 3 kilometers of cave. It's 500 meters deep. So there's a lot of things to be done and discovered, even in places very close to here, sulfuric acid caves. Also, Shuangedong, the biggest cave in China, there's a branch in there. The walls are completely covered with celestine and gypsum, and probably also here in some places the sulfuric acid has worked. So there's still a lot to be done. I want to finish my talk with dedication to Alexander Klimchuk. He passed away more or less a month ago. This is only a week before he died, so he was still an active caver. It's a very pity that he left us so soon. We know how important he has been for hypogene karst studies. It's useless to say, and to the speleological world in general. What we want to do, and we thought about this a week ago, I think, with Philippe Audrat, we are thinking of dedicating a special issue of the International Journal of Speleology to his life and to the hypogene karst, dedicated to Alexander. I think from up there, he will look at us and say, this is a nice idea. So we will be advertising this in the following few weeks, sending out emails. It will be guest edited by myself, Philippe Audrat, and Arthur Palmer, and we hope it can be out more or less at a year from his death. So at the end of this year, we want the articles in, so we will be in time to get it out by May in 2024. Thanks.
Thank you very much. Do we have perhaps a question? All clear? Oh.
Working in such sulfuric caves, do you need sometimes that you have to wear a gas mask or like that?
Depending on the season in which you go, we bring them in active sulfuric acid caves. In Cueva de Villaluz in Mexico, for example, they use this because there's a lot of problems. You can have very low oxygen amounts, so you need this. And if there's too much H2S, you can breathe this in and it becomes sulfuric acid. In Albania, I was there in March. In the winter, when it rains, the fresh water, which is less dense, goes on top. So it avoids that H2S, comes free into the atmosphere, so you don't really need it.
But it is smelly everywhere.
You smell it, yeah, you smell it. Oh, yeah. It's awful.
One more question, Jo. You showed all those chemical equilibrium equations, but do you know if there's anything going on with research on kinetics of those?
With the what?
On kinetics of those.
I'm not a chemist.
No. I mean, if you reviewed the literature, so probably you...
How fast it is.
Yeah.
It's very fast. I mean, caves are very rapidly forming, for sure. Much more than in limestone, for sure. But then there's a protective way of doing. There's gypsum being formed, so the gypsum then coats the limestone, and this has a big effect on it. But it hasn't been really studied.
Okay, good.
Thank you. I would like to invite Kaja Schuschmel to present her study entitled Submerged Isola Karst, Investigating Submarine Sulfur Springs. She's coming from Department of Geology, Faculty of Natural Sciences and Engineering, University of Ljubljana, Slovenia, and she is working in the field of geochemistry and environmental sciences. Please.
Thank you. So this presentation is about investigation of submarine sulfur springs in Isola
on the coast of Slovenia and the challenges we faced. Here is Izola, a small coastal town in Slovenia, and it is a little bit special in geological terms because you can see that the almost entire Slovenian coast consists of flysch in yellow, and the green is the limestone, which is stratigraphically below the flysch. So in the Izola area, the limestone peeks out from the flysch, and it represents the Izola anticline, which formed as with the Istria-Friulian underthrust zone. So that's where you see all the thrust faults in the area. This is a nice picture that shows the Izola anticline. Okay, so here are our submarine springs. They occur along the Izola anticline, which continues below the sea. And you can see the holes from which the springs emerge. Izola karst has some other interesting geological features with some kind of hydrothermal characteristics. There is also a terrestrial sulfur spring, which occurs under a building. We know of two boreholes in the area. One is with sulfurous water, one is non-sulfurous. And there is also a cave in Izola. It does not have a sulfurous water, but it is believed to be of hydrothermal origin. And there used to be shafts, Sveti Peter shaft and Izola shaft, which had sulfurous and warm water. And this water was once used for the spa in the 19th century. It was supposedly quite popular with the people from Trieste, but it operated only for about 20 years. Then it shut down. Probably it was not economically viable. So for a long time, these sulfurous waters were a little bit forgotten. But then in 2002, Jože Žumer discovered sulfurous submarine springs. And he measured the temperature in them to up to almost 30 degrees. Now let's take a better look at how this spring looks. They occur in these funnel-shaped depressions, also called pockmarks. The spring comes from the unconsolidated marine sediments that cover the limestones, and they are up to 11 meters deep from the sea bottom and about 40 meters wide. This one is the biggest. There are also smaller ones. Let's see how this looks live. This is a video, and the video starts almost at the bottom of the pockmark. You can see the white filaments, possibly sulfur bacteria. And you'll see exactly the point from where the spring comes. If you want to know more about the white filaments, you should ask the chairman. Yes, you can see the below. Okay. So yeah, these are our springs. And of course, we want to know more about the sulfurous waters in Izola, so we decided to investigate them. In 2020 and 21, we took some measurements and sampling. The divers took the samples with the syringes, and we did the measurements of temperature, pH, electroconductivity, oxidation reduction potential, and dissolved oxygen on site as soon as the divers brought the samples on board. And then we prepared the samples for the laboratory analysis. We quickly realized that sampling of springs at the seafloor is very challenging. The conditions are difficult. It's dark. It's a small point from which the spring emerges, so it is really hard to hit the right spot and deep enough. So electroconductivity and chloride ions quickly showed that the best samples have 30 to 50% of seawater. But I will still show you the results because we can still get some information. These are the average measurements of temperature, pH, dissolved oxygen, and electroconductivity. You can see the submarine springs in red. And we can see that even with mixing, they have higher average temperatures. And even in the colder months, we measured temperatures above 20 degrees sometimes. Well, the pH is not as high due to the mixing with seawater, but it's still it's lower than seawater. These are the results of isotopic composition of oxygen and hydrogen. You can see the submarine springs on the mixing line between seawater in the gray square and the precipitation stations in the blue triangles. There's also groundwater, non-sulfidic groundwater in Isola in the green square. And we see that freshwater component probably originates from infiltration of local meteoric water. But the Isola area is urbanized, and most of the precipitation water goes into sewers. So let's look at where the Cosina and Portorož stations are. One is here, one is here. So one is at the classical Kars at the northeast, and one is south of Isola in Portorož, near the limestones that occur along the Dragonia River. And this could be possible recharge areas because they're not too far from Isola. These are the results of the isotopic composition of carbon from dissolved inorganic carbon. And we can see, again, the mixing with seawater. But if we look at the terrestrial spring in the pink square, we can see that this water is comparable to other karstic springs, and it is likely in contact with organic matter. So all this time I'm talking about sulfur springs. So yes, these springs have a really strong smell of rotten eggs. And we confirmed the presence of sulfide with the help of our friend Dinesh from Budapest because they reacted with methylamine blue. So our noses don't lie. And also, we detected higher iron and manganese concentrations. So it seems that we have some kind of reduction processes. We tried to explain the source of the sulfur by sulfur isotopes in the sulfate. But of course, we got the marine sulfate. It is possible that the reduction of marine sulfate is happening, but this doesn't explain why we have terrestrial sulfurous groundwater that's not saline. So we think the better explanation is that the sulfur comes from the coal from the Liburnia formation under the limestones that occur in Isola. And there was also a mine of coal near Isola in Sečovlje. So based on analysis from this and previous studies, based on history reports and data from the geological mapping, we made a theoretical model of what's happening. Regional groundwater flow is suggested to originate from either the classical karst on the northeast or maybe from the same limestone formations that are along the Croatian border. And then the water flows deep under the flysch, where when it circulates in deeper parts of the formation, it heats up, and then eventually it emerges through the fracture zones of Isola limestones, and it is also in contact with the organic matter, that is likely the coal. And the sulfite is most likely the consequence of bacterial reduction of marine sulfate or organic sulfate. So what's next? Of course, we need to get better sampling methods. So if you have any experience, ideas, suggestions, I'll be very happy to talk to you at the coffee break. We need more terrestrial groundwater data, and when we get better and more data, we can make real geochemical models. If we do some microbiology analysis, that would also be helpful to understand the geochemistry of the springs and the ecosystem they support. Besides the ecological points of view, the springs are interesting because they may offer an insight into the geothermal potential of the area. So thank you. If you have any questions, and we can also discuss at the coffee break.
Thank you, Kaya. Do we have a question, maybe?
Maybe I can just speak up. Maybe it's a stupid question, but I noticed the depth of the spring was approximately 20 meters.
Yes, from the sea level.
Yeah. Have you considered installing some kind of buoy?
Some kind of a?
Buoy, like a float on top of the spring that's anchored to the bottom of the funnel, and that maybe has like a hose that goes to the bottom, and then you could go to the buoy and pump out water from there.
Yes, we consider this is one of the ideas, yes.
Thank you very much. I think with this we conclude the session on speleobiology and geochemistry. Just a short announcement from our technical support. For the next session, please upload the presentations. Thank you.
Hello, everyone. I have few informations about today and Thursday and also Friday excursions. So all posters are available for retrieval at the hall at the Institute. They will be taken down by the end of this evening, so please go grab them up. Then afternoon excursion will be similar as yesterday. We will leave at 2:30 from the bus station. We will be again walking a bit and taking the bus a bit, so please be prepared for both. The tomorrow excursion will, just a second. Yeah, we will depart at 8 o'clock from the bus station. Please be punctual because we cannot wait. The excursion will be a bit longer. And also the lunch packages, we will receive them after 1 o'clock. So you can also prepare some small bars or something for the morning so you are not hungry till the lunch. We will be both walking in the cave and outside, so also take a jacket or something that you're not cold. For tomorrow excursion, you can still apply. There are still some few places left at the registration desk. But the Friday excursion, I have the registrations for the Friday excursion, still some places. And the map will be here at the coffee break, here at the table, because we will close the registration room today. And by this registration form, there is also the book of signatures. If you still want to place a signature just for our institute to remember everyone that came to the Karstological school, we will be really happy. Yeah. So Cyril is telling me that today we will walk 6 kilometers, supposedly, so we will be walking. So those who, Yeah, so those who are not feeling the walking, it's better than you stay here in Postojna. Tomorrow at 7 o'clock will be the gala ceremony. Don't forget, at the Institute Hall downstairs. So, yeah, this. And also on Friday, the excursion will depart at 9 o'clock and finish around 5 p.m. And all the informations are also just in front at the door. There's a list of information. So if you have any questions, I'm able to answer. Just please find me somewhere. Thank you.
Paul Griffiths, heb je ook geen winnen? Ja.
Last locality, the material is so good that even ancient DNA could be extracted. Comparison possibility. Patella is definitely of rhinoceros, but we don't know. And the sediment is dated 150,000 years. Example of the departed bones. Excellent. We will collaborate. Well, fine. I have to ask another photo. Too much tricky. But we are actually looking for international collaboration. We are being old man as participate, but only work on him. He said it's very tricky to make some post-cranial determination. Especially we found but I compare, I have a comparative collection. I compare I will just check out if so we decided to be both beginners, but it's a colleague of mine, Koshka, from the Karstological Institute, he said would be also very similar. So whatever collaboration is no, must be about 500. Possible code. So carbon will not work, but I expect to hear about it. He made me the data for this locality. Carol 50 or 170,000. Hopefully, and now I want to spare some money. I will pay him to make another. For this we have oratorial notation, have one of the last one. I change a bit my field of work from business to paleontology. How is it? Така майка піча, я собі то знав. А слідно можливо бачити. Головний музей. Музей. Gremo še eno pozdraviti. Pa se popodne zvečer vidite.
So we are going to start the last session, which is dedicated to protection of karst. The first keynote speaker will be our good friend, Paul Griffiths. So the word is yours.
Can everyone hear me all right? Yes. Okay, I'll probably walk around with this thing. If you bear with me, I made some notes to myself that I must absolutely get out now, because if I run out of time, I won't get a chance to do so. I wanted to let you know I'm a product of the Karstology program here in Postojna. So I'm very thankful to all of the people from the Institute, because my topic was rather influenced by all the discussions we had over the years. And I must thank so many people in the room. Andrei Mihayuts, who's probably watching us, Nadia Zupan Haina, so many people. I couldn't possibly name them all. And I had a very fine mentor or supervising professor, Dr. Andrei Krantz. And he and everyone else at the Institute, if I could say one thing, they always injected humor in our discussions. So that's probably the only thing that would allow me to use a little bit of latitude and make this a little more of a fun exercise. The other thing I wanted to mention is I started, Francie contacted me and asked me if I could do this or would do this, and I said, yes, of course. And so I thought about it. It was about 6 months ago. And I thought, well, the easiest thing to do is to write it out. So I ended up writing about 80 pages. Now, now I'm going to use none of it, because over the last few days I've had to constantly recalibrate, readjust my slides because of the many fine presentations that were done, especially on the progression of karstology as an interdisciplinary field, and also the progression of conceptual modeling for karst and so on. My pilgrimage to Postojna was 1977. I drove a two-cylinder deux chevaux from France, but couldn't get inside the country, so dumped it in Trieste and took the train. But back then, and this is another point I wanted to get across, was I was very speleocentric. I was a caver. So I wanted to mention that because I'll be talking about speleocentrism as one of the barriers to making progress and advocating for karst. So you could say I was a crack addict, not cocaine, but rather I was addicted to caves very early on in life. And so when I came to Postojna, I naturally went to the cave and probably walked blindly past the institute. So it was only later that I came to realize I needed to get away from caves only and think more broadly. Right, so it's relevant to this discussion possibly. And the other thing is a couple notes. One is, like I come from British Columbia, and nothing special with that. It's just that in that Canadian province, we've defined karst landscape very broadly. In other words, it's probably the broadest interpretation possible. We would view the landscape as including the vegetation, the soils, everything. In other words, the biosphere, the hydrosphere, the lithosphere, the atmosphere, everything counts. So so when I refer to karst, I'm thinking the karst landscape system. The other thing is, legally, we define a karst system as including contributing non-karst drainage basins. So for us in our Canadian province, we view the karst system as including non-karst terrain, if you like. So it gets a little confusing for some people. So I just wanted to say that. Yes, so after listening to everyone, I rejigged my presentation a bit, juggled some slides. If I don't get through them all, I'm still going to try and get this done in 30 minutes allotted to me. That's no problem. You know what they say, you're born with a brain, but it stops working the moment you do a PowerPoint. It's probably worse for a keynote. I've never had 30 minutes to talk. Usually it's 12 and 15, and I've been known to go over. So imagine the risk when you have 30 minutes. But anyways, I'll activate the next slide. Right, so briefly what I propose to do is to give a brief overview of where I'm from, because everything I say is mainly about my region, because I wouldn't purport to give you examples and then pretend that they might apply to your region. You'll just have to look at what I'm saying, and maybe you'll find some things that resonate with you in your particular region. But I don't want to be so presumptuous to think that I know what's going on in your part of the world. I'll cover a little bit the driving force behind sustainable management of karst ecosystems. I won't dwell very long at all on the history of karstology or the development of conceptual models for karst, because so many people have spoken to that. But I will talk about some of the issues and limitations or barriers when advocating, in my region anyways, for a broader concept of karst, a more holistic and ecocentric, if you like, meaning for karst. And then I'll try and end with a summary. Basically, I can tell you what it is now. It's just carry on doing what you do and what you do best. We obviously need international collaboration, and I think we've got it, especially with the IKSs that we have every year. Sorry, I'll move forward here. Now, I asked myself, why on earth, what compelled Francie to ask me to do this? And then I thought more and more about it. It's because he spent 2 weeks as a prisoner in my car last year. And I tortured him with stories, and he probably thought, I'll get him back. I'll get him to talk about this, whatever subject he wants. I think that's the reason. And right off the bat, I'd like to absolve myself of any responsibility for perhaps insulting people and blame it on the organizing committee for the International Year 2021. In other words, what I'm trying to say is I might be saying some things that suggest that some people might have a less than full understanding of karst, not in this community, but in government and in industry. And I don't know whether we're live streaming or not here, but I'll be very careful about what I say. So briefly, I'm from Vancouver Island on the Pacific coast of Canada. We've got a lot of rain. About 4% of the land surface area of Vancouver Island is karst, mainly high purity limestone. Forestry is our main industry, and forestry has degraded a lot of the karst landscapes, right? And although we made progress, especially with the help of Derek Ford, we prepared many guidelines and protocols for managing karst as sensitively as possible, and many of them were adopted. The problem was in the early 2000s, the government changed the legislation and went to what is called results-based management of forests, results-based. Now, I'm going to hopefully provide a few interesting asides. I remember I was asked by a session chairman of the Geological Society of America to go to Denver for an annual meeting to give a talk on the experiences in British Columbia with managing karst in a forestry context. So I said, sure, I'll do it. The title of my talk was, Is Self-Regulation Working for Karst Management? So I was about to give the talk when someone said, what are you doing? You don't need to give the talk. We already know the answer. The answer is it can't possibly work. That's why we have laws to prevent people from murdering people. So I gave the talk anyways, but anyways, Vancouver Island is very wet. It used to be fairly well known for pristine karst landscapes in association with primary old growth forests. Not so much anymore. Why? Because the timber was valued as a commodity, and much of the island, maybe more than 70% now, is clear cut. And Derek Ford well knows what happens when we clear cut the forests on Vancouver Island. Very early on, he came to Vancouver Island and he brought graduate students that studied the problems. As early as 1986, the results were in on what to quantify the soil loss and other problems. Believe it or not, in my part of the world, there are political campaigns against karst. Now, unfortunately, I couldn't find the video from the 1980s where there was a rally. It was shown on TV, in the TV news, and there was a placard that one of the protesters was holding up, and it was exactly like this. In other words, it was an anti-karst, if you like. It was probably anti-cave statement, political statement, that no, we could not protect and conserve this resource because it is in conflict with resource extraction. So that's a bit of the background to the tension between environmental management and industry. Just a little bit of the tension there. So again, in the early 2000s, oops, okay, they removed what is called the administrative burden. I don't know whether you would understand it, but the red tape. In other words, in a results-based regulatory regime, you remove the rules and you simply ask for a result and you trust the proponents of activities, land use or resource development activities, to do the right thing. So in exchange for that, there is the freedom to manage so long as you achieve the results. But people have to be watching to make sure the results are achieved. So again, we had some big timber, and this will become relevant later, but being very large trees and heavily branched, obviously there's a lot of breakage and a lot of fuel loading on the ground, which becomes a factor for fires. Now, a quick aside, here's the contrast between Slovenian forestry and British Columbia forestry is simply this. In Slovenia, the policy is to mimic nature. In British Columbia, forest professionals believe clear-cutting mimics nature. That's basically it. So that's the difference. Now, as another interesting aside, you may not be as familiar in Europe, but Rachel Carson was a well-known author. I think she published this book in 1962. And it's seen as a milestone in terms of the inauguration of the environmental movement, at least in North America. Now the thing is, and I mentioned this at the beginning of my thesis, but now I know more about it since I wrote the thesis, and that is that there was a spraying program on northern Vancouver Island that killed all the fish. But precautions were taken because they had experience with killing fish on the east side of Canada. So they instructed the pilots to spray one wingspan away from all lakes and streams. The problem is, on northern Vancouver Island, there was a lot of karst. So you can imagine, like, it's a perfect illustration of the sensitivity of, or the uniqueness of karst systems. So if ever you read the book, you'll see that the introduction is dedicated to that. This was DDT spraying, or Agent Orange, basically. Killed all the fish and aquatic insects. And I've interviewed people who were there at the time. This is in the early 50s. And they say there were so many dead crustaceans on the beaches, they would walk deep, deep into them, halfway up the shins, like halfway up to the knees. Right? And I've since learned that the Central Intelligence Agency in the United States tried everything to discredit Rachel Carson because they regarded it as a very dangerous book and a very dangerous movement. It was 1962. Sorry to go on about that. So it's the same talking points here, still on Vancouver Island. Forestry is one sector. It has by far the greatest footprint on the landscape, like on an area basis. Now, what's the driving force behind doing anything? Here, I'll mention Derek Ford again, because he was invited to this conference in 1997, and his dinner talk was titled The Limestone Imperative. So you might as well say Karst Imperative too. And just as an interesting aside, notice this is the 1997 Karst and Cave Management Symposium. Up until that year, all of the symposia were National Cave Management Symposium. But the Americans were kind enough in this first joint US-Canada symposium to change the name to Karst and Cave. And I think it stayed there. And not only that, but that year the theme was highlighting forest karst ecosystems. Obviously, the other imperative is just the global distribution of karstic rocks or karstifiable rocks. You know, we hear different percentages, but let's call it 15.2% for the sake of this map. Obviously, with such a distribution, it figures very prominently and hence feeds into the imperative to manage it wisely. I won't dwell on the progression of karstology. I had originally planned to, but obviously many, many disciplines are involved with it. I don't know whether Pavel Bosak is here, but we were talking, I think it was last year or the year before, about the silo effect. People are working in specializations. And he was telling me about the concept that we have too many specializations. They're like branches of a tree that grow further and further apart. So there's that kind of issue when you have so many related disciplines and subdisciplines. Of course, along the way in the development of karstology, there are any number of books. I just happen to use these two because Paul and Derek are right there, and they're the authors of these books. Right? So there are many books that support and give recognition to karst science as a legitimate science. I can just skip over this, but I just picked 3 things along the way. One is the recognition or the greater recognition of epikarst. Tanya Pipan, if she's here, is one of the people who wrote about the promising habitat of epikarst. The use of vulnerability mapping, obviously, Natasha Ravbar and her colleagues were involved in the development of modeling for vulnerability of groundwater, and the increasing attention paid to microbiology, the biosphere part of the karstosphere, if you like. So we don't have any figures for that. And someone pointed out to me, I think it was the person who took that picture, Penny Boston. She said, do you realize in a single drop of trip water, there are 160,000 this and 200,000 that? And so her point was scale as well, scale, right? So with the work in microbiology, this is certainly an area of rich research. I noticed that the progression of these things, and I should move more rapidly on this, is reflected in the schools, right? We learned the other day, the first one was in 1993, and happened to highlight the KRAS itself. Right? So I took a picture the other day and then realized, well, the first school was not held here, but in a town that's behind the viewpoint of the drone here. Right? That was the first one. Right? I won't go through them all. But you know, the second one focused on the KRAS region again and the science of karstology and how people perceive karst landscapes. And I believe, I did not attend that one, but I believe there was a woman who canvassed the room and used the participants in the school to give her information on their perceptions of karst. And then she was going to go on to do it with people who lived in the region and that sort of thing. Very interesting. And the next one was the that I'm picking out is 2006 Sustainable Resource Management for Karst. 2017, one of our friends, an Indigenous person from the west coast of Canada, actually came to Postojna and presented on an Indigenous perspective on karst. And a funny one was last year, one of my compatriots from the University of British Columbia, an architecture student, gave a poster presentation on the influence of karst on architecture. Let's just skip through this a bit. You all know about early depictions of karst systems. Moving fast forward, I particularly like this one, which concentrates on the basic environmental spheres, if you like, the primary environmental spheres by Andrzejczuk, who I believe works in Poland but is from Ukraine. And so I kind of adapted his diagrams a bit here. He added the anthroposphere to the four ones that you usually see, the biosphere, the hydrosphere, the lithosphere, and the atmosphere. And his point was that if you look at the relations and the number of elements, when you're dealing with a non-karst landscape, there are relatively few of them, like 10, let's say. But once you go and combine with the subsurface, then you have many more relations. I think it's 45. Right, so his point was things get more complex with a karst system. But the model was dimensionless, didn't favor any particular environmental element. Here is, we adapted this in British Columbia when just talking to people, a sort of a spherical model, loosely based on Andrzejczuk's as well, but splitting the spheres into more and more spheres, like pedosphere and flora and fauna, as opposed to just total biota, and so on, just to get the concept beyond caves as well. There could be future enhancements to that model. Now, what's the problem? The problem is the public understanding of karst lags behind your understanding of karst, right? So that issue is not in line with the karst imperative we spoke of, right? And some people responsible for managing karst, they have no understanding of it, right? As the organizing committee recognized for the international year, and they may actually actively oppose sustainable management of karst. Right, so that leads to harm to karst, right? And that goes against the limestone imperative. So there's a limited framing of karst from some quarters that serves whatever purposes, and that undermines efforts at sustainable management of karst. The other day, I ran into a group of students who encountered a forest company representative who proclaimed there is no such thing as a karst system. So I don't know what the point of saying that to the students was. But it was rather interesting. So some of the barriers, very quickly, I would list them like this. I don't propose to spend too much time on it, but one is what I call the mineral shell paradigm. It's the belief, well, all that matters is the rock. If we don't damage the rock, what's the problem? Right, so it's a little less ecocentric approach to karst management. The other is the appropriation of karstology by false experts, the dissemination of misinformation, the race to the field mentality without an adequate grounding in karst theory. I got a funny one, is karst truly cursed? And shortage of karst education in general. So the mineral shell paradigm is depicted this way. It's basically people who regard karst as just the bedrock shell, so nothing else really matters. It's pretty durable, so what could possibly go wrong? That influences environmental risk assessments. Forest professionals in my part of the world do not regard the trees nor the soil as part of the system. So obviously that serves their purpose well. If we apply the mineral shell paradigm to a simple doling, obviously the result for a forest professional might be acceptable, but the result for another person, a karstologist like Andrea Hayes, might not be so acceptable. Right, there seems to be no understanding. It's like evapotranspiration doesn't enter the picture anymore with many forest professionals, and yet did they not learn this at university? I don't know. The mineral shell paradigm treats the doling, or at least the dissolution doling, like this. That's it. So as long as that's not damaged, we're okay. There's no recognition of the ecological significance of dolings. And of course, everyone's frightened because all sinkholes are dangerous. The MSP, or mineral shell paradigm, applied to fire impacts, well, no problem, right? Because again, the forest vegetation is not considered a component of the system. So you can have total loss of soil. There's abundant fuel because of the breakage of large trees falling on the ground. So these uneconomic pieces are left on the ground and they provide the fuel when the humidity is low and there's a condition source. Speleocentrism is a barrier of sorts because it's very convenient to focus on the caves and to focus on their use for sport and tourism as opposed to managing the overall system. Not helpful is promoting adrenaline-fueled experiences with caves. So I don't know. Personally, I think cultivating a fascination with caves is just not particularly helpful, at least in my region. And it's not like people treat our caves with any respect. And this cave is in the highest class for provincial park possible in British Columbia. And of course, ESAN doesn't help by combining the adventure of caving with the purchase of one of their SUV vehicles. The forest professionals, maybe they could be forgiven for thinking that karst is all about caves, because in their own reference materials, karst is depicted as being equivalent to limestone caves in writing. So you have forest professionals thinking, oh, well, you know, we're reducing karst management to cave management. The appropriation of karstology by pseudo-experts is not particularly helpful. Usually the pseudo-experts like to say, oh, well, in other words, it feeds into the many different opinions fallacy. Right? So, oh, there's many opinions about karst, therefore, how could we possibly arrive at a simple conclusion here? That works very well. The dissemination of misinformation. This is a problem because of Brandt-Levy's law. It takes 10 times more effort to correct misinformation. Like, I'm too polite to use the actual language for the law, but basically it's a lot of trouble to reverse bullshit. My colleague Carol and I have taken to traveling around and presenting the 10 top misconceptions about karst in British Columbia. That became karst and karst management in British Columbia. She doesn't look particularly happy. Usually she's not happy when we're doing this because we get such stupid questions. Our own government constrains the definition of aquifers, basically to exclude fissured or fractured bedrock. Our government suggests that karst purifies water. They actually put out this information. The idea is that it's a natural purification mechanism, so we have one word for that. It's Walkerton. In the year 2000, 7 people died and 2,300 people became sick in our province of Ontario. And then we have the government proclaiming that, and rest in peace, Alexander Klimchuk, that all limestone caves are caused by volcanic activity. Right? They actually, the thing is, public funds are used to disseminate this information. That's exactly what they say in literature paid for by the taxpayer. The race to the field, no problem with going to the field. It's just that our experience is people have very short attention spans, especially younger people. Yes, they'll take a selfie, especially if there's a cave entrance. But in terms of an adequate discussion of the principles of karst, no. Selfies take precedence. Now, is karst truly cursed? The thing is, this is a piece of proposed legislation. It's serious legislation. But karst was spelled K-U-R-S-T. Okay, once, no problem. But this was done in 2 successive years because the first legislative bill died because of the scheduling of the legislature. So it happened twice. So what are you going to do when legislators can't spell terms in legislation? The shortage of karst education, we have no dedicated karstology program anywhere at our 4 major accredited universities. So while there's an enormous department of forestry with a staff of 190 at one university, the University of British Columbia, and these people become decision makers and policy makers and influence everything about karst, there is nothing for karst. In the high schools, there's a little bit of education in Ontario, Derek's province, grade 11. In my province, there was, and then it disappeared and was never replaced. So all of these barriers can reinforce each other. And so Carol and I coined a new sub-discipline of karstology. It's called impedimentology, which are barriers to making any progress in karst. Right there. I guess we should think very quickly about the audience. You know, who is the audience we're trying to communicate with, and how do they receive the information? Because it's not like the old days. You know, they're looking at their phone, and what is the validity of website information? Anybody can mount a website. So that's an issue. Obviously, there are challenges of conveying karst itself, everything from 2D cutaways. We talked about that the other day, to 3D, to 4D. Everything's possible. The Avatar movie itself was largely based on karst of Thailand and elsewhere. I actually talked to the studio man about this. It sounds crazy to mention it here, but when you consider how many millions of people have seen the first movie and the sequel, there might be subliminal messaging that's possible to convey the complexity and the diversity of karst systems somewhere in there. Now, this is the end. So a couple final words, and one is a word about the role of indigenous peoples. Because 6% of the global population, but they own or manage or have an interest in about 32% of the global land surface. And if you look back at the map of karst distribution and you look at their distribution, you can see the overlap. So they're obviously a factor, and the IUCN guidelines recognize that, but so does the United Nations. And the new guidelines obviously cover that. And I noticed, by the way, in the guidelines that one of the guidelines is not to cut natural forests. I wonder how many people have thought about that one. Not to be clear cut, not to be logged or subjected to any human impact. This is natural forests, right? But we still have natural forests here and there. And a word about advanced karst education. I can't say enough about the usefulness of the program here. Can you imagine like having a doctoral program in karstology? For one thing, recognizes that there is such a thing as karstology. That's wonderful, right? That's fantastic. So that's what we need to do is carry on with that and carry on with the schools, right? So obviously, I would just end by saying we just need to keep doing what we're doing and just persist. However cynical we might be from time to time, it's a good thing that we talk together every time we meet here. Everybody is just so wonderful, and everyone is contributing to an overall goal to deal with the limestone imperative. So thank you very much.
Thank you, Paul, for your enthusiastic point of view. It is always addressing us, and I hope we will have some question. Okay.
Thank you for a fantastically nice talk. And I think I have too many questions, but I have a comment to you and to the organizers, because I think the videos on YouTube from this is as fast as the talks. And I think if it would be possible to post the PowerPoints themselves on the website so we can have time to read all the text, that would be very good. But I mean, all my compliments to your talk. It was very, very interesting, and it's immensely important.
Oh, well, thank you, Stein Eric. That's much appreciated.
I think that it's not a problem if you agree, actually, the lecturers, so we can arrange this. Some more comments? If not, we will move to the next presentation that we will be given by Didier. We will move to France, but we will stay within similar topic.
Hello, everybody. Thanks for the Institute to have the opportunity to present this project. We have done in France since 2 years now. It's a common project with scientists, speleologists, and some cave management for cave bats habitat in the Jura Mountains. And it's a project with the support of the region, Bourgogne-Franche-Comté, in this area. Yes, the questions say how a management plan for a network of bat cave can stimulate cave research and also can help to have a better conservation of the cave and also for the bats and the different cave ecosystems. Yes. These networks, we are in the east part of France, in Jura Mountain, and there is 7 caves, very small cave. Yes, it's very small cave because most of them have maximally 100 meters long of development. They are situated in the 2 parts of Jura. Most of them were at the eastern part of the Jura, and some of them are at the Plateau de la Saône, the starting of the Bassin Parisien. There is a river in the two systems, the Saône and Onion River, with the boundary of the two systems. These caves are known for a long time because during the 19th century, for example, there are many studies for archaeology, paleontology. It was also described as karst heritage for Pliocene and Pleistocene period, and also it was known for some of them for paleoclimate environmental records. And this is very interesting karst and cave landscape. So the management, done by my colleague, Catherine Dioniso, have questions. They start a management plan for 2018 from 2023, and they try to have the better accuracy to understand what's happened in the cave and what is the state of the conservation for bats' habitat. So they used the national management plans who are in France in the frame of Natura 2000. And there is some different actions, yes, to understand the bat populations, to understand the bat environment, and also to make some didactic actions about bats and also cave. So yes, we have reflections about that, and there is some questions for the next management plans. Yes, because there is a decrease of bat populations, biologists and cave managers have monitoring that. And the questions, what is the reason for that? How the bat biotope evaluate? And for that, we decided to create a group with biologists, ecologists, and karstologists. And the question was, yes, okay, we want to understand a biotope, but what are the characteristics of this cave, of the reserve? So for that, we decided to have collaborations with karstologists, speleologists, archaeologists, and also paleontologists. And what are the characteristics of the bat habitat inside this cave? Yes, and for that, yes, we have another group. So we decided to organize all that and to practice on the field. I'll present briefly, yes, what we have done for the geomorphology, because I'm geomorphologist, and what we have done, what the plan we have done, and what the value we have tried to evaluate and to take in account for that. The K lecture was the cave landscape. You have an example from Coliboia Cave in Romania, because it's very, very representative of all that. Yes, what we see when we arrive in a cave gallery, and what we can observe, and what are the phenomena in functionally in this gallery. Weathering, condensation corrosions, notches, bat polish, guano, speleothems, sometimes rock art or archaeology. And yes, and we try to make an inventory of that and to make the description of the natural and the cultural heritage and what are the dynamic between all this feature. Also, with biologists, we have made the inventory of the bat colony, the present day's occupation by the bat colony, and in many of them, we have seen it's very, very small quantity of bats. Yes, for all the reserves, it's less than 4,000 bats in all the reserve. And for example, in Grotte de Chensey, it's only 200 for nursery. And yes, during winter, it's 10 or 20. In Grotte de Beaumont, it's a little bit more, but it's less than 500. Rivière de la Baume, it's practically nothing. Baume Noire, a little bit more. And for example, in Grotte en Y, there is no bat presently. So, when we observe the landscape, the cave landscape, we can see that there is some old occupation, some indications of old occupation, because we can see many features of biocorrosions described by Philippe Audrard and so on since this time. And we can see, yes, there is some cupola due to the guano decompositions. There is some accumulations of guano. There is some notches. There is, in different places, some yes, fruit or tune or some rocks falling due to the guano corrosions, and sometimes a completely reshape of the gallery patterns. So in the past, there was a lot of caves, a lot of bats in this cave, but actually less and less. And we try to discuss about that. Also, for the other information we have in the cave was the condensation corrosions effect and what is the actual climate in the cave. It was really easy in some cave, especially in Rivière de la Baume, because you have a Bajocian limestone, sorry, with pyrite. And yes, in the part with cold wall and cold air, you have many growing of gypsum crust, very important growing of gypsum crust. Sometimes there are 20 centimeters of gypsum, and at some period, especially in summer, or when there is some, when the bats are present, there is warm air in the upper part, and you have condensations of the wall, and you have the complete dissolutions of the gypsum. And you can see on this photo, it's really clear the limit of the climate regime in this area. In Grotte de Chensey, for example, you have many places with condensation corrosions, and after you have calcite due to the condensations. And it comes over some inscriptions, and some of them are from the 18th or the 19th century. And you have the growing of small speleothems on the wall. And in this area, there is a disc growing due to the condensations, and yes, falling down due to the too big growing. And under we have in this, yes, a painting and with charcoals, and we have a dating with archaeologists of this, and it's 8,000 years old. So we can see, yes, the activity of the climatology in this cave. We have tried also to make measurements, to have a network of measurement of the temperature. We have not the result for this moment, because we have started only at the beginning of the year. But this is an example we have done in Mas d'Azil cave in Ariège, in Pyrenees. And we have used, yes, this mat, and we have put some loggers in different parts of the gallery where there is some bats. And we can have some very good result because we are able to see when the bats are present in the gallery and when they leave the cave. And what is really interesting in this cave, especially in Mas d'Azil, you can see the temperature during the year at the bottom at this level, and it's very confined gallery at the bottom because the temperature is very, very stable, less than 0.2 degrees of oscillations. And when there is the bats, you can see you have an increase of the temperature because we are 12.2 degrees when the bats are present. In another cave, in Espelin Cave, yes, it's a place where there is 2,000 bats for nursery, and with temperature monitoring with this system, it's a huge gallery. It's 12 meters high, 20 meters large, and it's in the Pyrenees also. And in this, with this system, we can measure when the bats are present in the cave and when the new bats are arriving and growing and after leaving the gallery. So it's a very good system and very precise. So we have decided to install that. With yes, we have installed 40 loggers in the different cave to have this measurement. The problematic of the condensation corrosion is also for the preservations of paleontology. You have some bear's claw marks here, and due to the condensation corrosion, years after years, they became more and more discrete or recovered by calcite. And it's another problem. Now the cave is closed because many people come and break the flow stones to access to the layers under with clay and to find bones remains from bears, speleo bears. So we have decided now to close the cave with a system for the bat circulations. Okay, so there is also an important thing because with this cave, due to these observations, it's the evolutions of the cave in the context of the Jura mountain, because we were wondering what are these small caves done in this area. So we have reflections about the long-term evolutions of the Jura and the positions of this cave. For example, in this, if we go in the Rivière de la Baume valley, it's close to Poligny, and there is a pocket valley, is Reculé des Vaux, and there is an anticline. This is Jura Plateau, and this is the limit of the ice cape during the last glaciations. And the cave system is in this area in yellow, just at the border of that. And inside, we can observe the incisions of the river through the times with a very small basin of the cave. And for that, with this analysis, yes, we can see this this cave is completely parallel of the pocket valley. And at the beginning, it was one of the part of the evacuations of the ice cape. But the main drainage was by the pocket valley, and the other main drainage was by the northeast to another cave system, Arbois cave systems. And this cave, Rivière de la Baume, has not evolved so much because there is the difference of the two drainage. For example, another in the Doubs Valley, close to Besançon, you have Creux à Pépé, very, very small cave. Yes, it's 10 meters long with a small pitch. And there is some bats in this area. But at the beginning, there is a lot of bats in this area. But now only, yes, 100 and less in this part. But what is really interesting with this cave, it was the Doubs River, like this, making meanders. And there is huge differences between Grotte d'Ossel, the actual active systems, and the older karst organizations. So we can have many dolines linked to the Ossel galleries, and it was an evolution of that, and now the springs are in this area. So it's important to see that this small cave is not just a isolated perch cave, but it was an old grit system. Also, in the other part of the Jura mountain, there is huge systems near Baume-les-Dames. This is Vallée de l'Oignon here, Vallée du Doubs here, and we have in this plateau all the organizations of a cave system with a very complex network of different caves. Actually, the bats are only in this small gallery, Grotte des Ours, and the grotte en Y is closed. There is no bats actually. And there is this cave, Source de Gondenan-les-Moulins, and it's important because actually there is some bats arriving in this area. And we have decided to understand what is the organization and what is the ecosystem of Rivière Souterraine de Surrey. And for that, we have put some loggers to monitoring the water and also make some dye tracing. Also using some stalagmite to have some paleoclimate reconstruction and dating to understand the cave evolutions. And in Carrière de Romain, there is also a very important paleontological site with a lot of fauna from the cold period of Pleistocene. Grotte du Croteau is also a sinkhole, and we have made some dye tracing from here, and all arrive at the source of Gondenan-les-Montbis. This action was led by speleo and also hydrogeologists. And we make some, reconstruction of that using model, and also we have put all the dye tracing axes on the geological maps. So it was, a good action. In the Plateau de la Saône also, we have a cave perched over a plateau, and it's a big gallery, but completely isolated, and this cave is completely disconnected to the other part of a karst. It's also an old phreatic gallery due to the Saône River, but a long time ago. And we use also some geophysics in this part to understand if there is some continuations and if we can find some archaeological remains, because in this part there is an important archaeological site from the Upper Paleolithic. So in Plateau de la Saône, we have made also another small cave, grotte. Okay, the small scale, the grotte of Echenot-la-Méline, and in this there is a lot of archaeology since the 19th century with a very good documentation. Important work from Michel Campi, who has identified the different period of the Pleistocene with some fauna, with human occupations, and also all the evolutions of the valley during the Pleistocene. It's also an important site, very close to Vesoul, because here there is a very important karst system, Fond de Champ-d'Amois karst system. It's a long river, underground river, who is the water supply for the city of Vesoul and all the environment. And the dye tracing have shown that there is an evolution of the Méline Valley to the Columbine Valley, and now it's only this small remain of a cave. So also in Beaumont Cave on Plateau de la Saône, yes, you have a huge gallery here, completely perched on the plateau, and when we make some reconstructions, we can see a past water table at this altitude, and after the evolutions with the Onion River flooding down 70 meters down actually. So, to conclude, this participative project, yes, with volunteers, managers, speleologists, cavers, provide new data and observation for the cave and their environment. We have some new descriptions of that, a better understanding of the feature in the cave, some geophysical model in that part, and we have started to have a very good monitoring of the actual climate in the different cave. It's the opportunity to have some monitoring sheet with in the different cave the most significant feature and what does it mean to help the managers, but also the politicians to organize some project to continue to study. For the land management also, because in some place, for example, here in Beaumont, we can see that a part of the growth is outside the perimeters of the reserve, so it's necessary to adapt it. Also in Shenzhen, a part of the growth is completely out the surface of the reserve, so it's necessary to extend it for the better protections. We have a tool for management with GIS, and this permits to have a very good representation and understanding of the evolutions of the cave in the Jura Plateau. So with that, now we will try to find new projects to have a better accuracy and a better understanding of all of this. Thank you for your attention.
Thanks for presentation. We don't have time for questions, so we will follow by the next presentation given by Francesco Zauro.
Okay, good morning, everybody. First of all, I want to thank Franci, Nadia for inviting me, for all the Karstological School organizers. I want to say that I'm also kind of a byproduct of the Karstological School because I came here with my father when I was a child. I was very speleocentric, not very interested in karst, but more in caves. But then, thanks to many years of listening about karst, also I got in this kind of research. So I will talk to you about planetary caves and what they can tell us and what is the perspective looking farther from this planet. So as a speleocentric person, my question is not just what is out there, but there are caves out there in the universe and also karst, of course. And let's say that this just 50 years ago, this was kind of only related to films, movies like Star Wars. But already in 1969, while the first men were landing on the moon, we had some ideas, especially for the moon, about the possible origin of lunar rills, these kind of narrow valleys related to lava tubes. And it was Greeley, the first one to talk about that. And then we had other publication, Hartz, in 1985. But also in 1978, you see, there is on the proceedings of a Congress in Italy by Castellani and Cigna, they were the first really to postulate about the potential presence of karst on other planets. Unfortunately, it remained in Italian, and it was published just only, I think, last year, the proceedings of this conference from 1978. So let's say that up to, very few years ago, caves on other planets were just this, okay? Star Wars, and even think about life in these caves. It was quite interesting because it was already the idea of astrobiology in caves. But now, just in 2007, this started to become kind of reality when Cushing published this paper identifying 7 entrances on Mars through TEMIS images. Of course, this was possible due to high resolution images coming out from space missions. And in 2009, Haruyama from Japan described the first entrances also on the moon. Just consider that all these scientists were mainly astronomers, or Cushing is also a geologist, but let's say that they were not focused, of course, on cars or on caves. They just saw these holes on the planet. So now we know that these caves exist. There could be even something much more interesting. And so the research started. But before to deal with what we know, there is a big question, what is exactly a cave? And especially when we talk about planets, this can have different meanings. Of course, a cave is a space, as Carl said, rather than an object, and consequently its definition involves the specification of its boundaries. There are people even claims that an ocean below a crust, icy crust of a Saturn moon could be a gigantic flooded caves, but this is quite relative. So we, at least I am a speleologist, so my main definition is a natural underground opening rock that is large enough for human entry, but if we think about life and other living forms, even just very small pore spaces in a basalt are already caves. And of course, there could be also cast-oriented definitions, but let's say that if we want to think about caves, karsts, or let's say processes creating openings and voids below the surface of planets, we have really to completely make a change of perspective, open our mind, and think very, very broadly, because the process could be very different, and I will give to you some examples. So the best way to think about subsurface connectivity in other planets is to think about processes, and we can just divide in three main, let's say two main realms, which is we take out something, so the structural processes, which is karst is one of those. We are taking by dissolution some rocks and we are creating the cave, but it could be just erosion, melting, mechanical movement. And the construction also, we create something around this new cave that is a constructional process. So, for example, chemical deposition, but could be also mechanical accumulation. For example, about boulder caves formed in talus, and solidification, which of course are lava tubes, for example, on Earth. This kind of classification, I think it's quite useful when we think about not just only the process on our planet. This is another scheme that was proposed by Boston in 2004, was one of the first one, very, very efficient and dividing into different process type caves, solutional, erosional, tectonic, subfusional, and phase transition and constructional, and looking also to which kind of this process could happen on other planets. So on Earth, when we talk about caves, of course, we link it a lot to karst, because karst is the main process on Earth which is creating caves. But this is a very, very Earth-centric perspective. Okay, so the fact that we have created the term pseudokarst is exactly showing to us this Earth-centric approach, because we tend to describe as similar to karst what is related to other processes, but elsewhere, for example, if we go on Pluto, we could say that Postojnska Jama, it's a pseudo-cryovolcanic cave. So it's very, very human and Earth-centric. And I will show to you also an example which is Titan. So Titan is one of the biggest moons of Saturn. So to have the solution, to have a karst process, we need to have a soluble rock. Of course, on Earth, we have vaporized carbonate rocks, salt, and so on. But on Titan, we have rocks which are made of hydrocarbons, solid hydrocarbons. Then we need to have a solvent. On Earth, of course, this is water. We have an hydrosphere with lakes, rivers, streams. On Titan, we have liquid methane. And liquid methane is dissolving the solid hydrocarbons. And then we need fracturation. We need initial structural or textural rock, let's say, specific characteristic, like fracture that allows the solvent to get underground. And of course, on Earth, we have plate tectonics, which help us in this. We're just breaking the rocks and creating a network of cracks. But on other planets where we do not have plate tectonics, we could have just, for example, tidal forces. Imagine Saturn, a giant planet with a small satellite like Titan, and tidal forces are creating fractures. So we have all the ingredients in a completely different settings. And in fact, probably one of the places in the solar system where we have more cars is this small satellite. And there are publications from Alaska. They have been working a lot, thanks to the Cassini mission, showing the presence of thousands and thousands of odo lines, even structures that are looking similar to polye, possibly cave entrances. And in 2027, there will be the Dragonfly mission from NASA. So they will land with a drone on Titan. They will be able to fly. They are thinking even tens of kilometers around. And so we could have the first images of potential karstic caves in other planets in a completely different settings than here. So, of course, in other planets, the most important process probably is not cars, but is phase change, okay? So solidification or melting. This is usually what we know here on Earth about lava tubes, magma cooling down, but imagine liquid water which is coming out on the surface of a planet which is well below the solid line. So you could create also cavities by construction of ice on the surface. We have something like that in Antarctica. If you think about Mount Erebus, when the vapor is coming out, then it's solidifying in ice, and you can create this chimney which are actually empty. But also you can have melting, and we know that because of ice caves on Earth and sublimation, and there could be quite incredible caves due to sublimation around the solar system. And we have even images of those caves on a comet. The Rosetta mission from the European Space Agency. So a comet is made of ice, but also organic carbon molecules and so on. And they saw this kind of pits. And looking to specific, let's say, stretching the illumination of the image, you see these jets of vaporized material from the comet which are coming out. So there is even kind of expulsion of this sublimation effect and creation of voids. And this is why the comets are also consuming a little bit from the inside, not only from the surface. So you see that perspective can change a lot if we look to other planetary bodies. So when I started to study geology, because I was passionate about speleology and caves, I never thought before that I was going to teach then planetary geology at the University of Bologna. And I think planetary geology in general for all fields of geology is very, very important discipline because open our mind and way of thinking. Because if we look to different planets, we understand the importance of planetary parameters. So if we change, these are the rocky planets of the solar systems, okay? So we have more or less the same density, same type of rocks and materials. But if we change the dimension, we change the gravity, then everything will change also in the processes that we can find on these planets. If we have an hydrosphere, also it will be very different than a very acidic planet. The surface temperature can change a lot, and we know that surface temperature can change also solubility for different kind of materials. The geodynamic history, there are planets that had plate tectonics, others not, but have other processes that let's make them kind of alive, and of course life. So without life, we would not have carbonates probably in this planet, and so we would not have cars, or very few, or very different cars, maybe evaporites. But life is also one of the very important things. So thinking about other planets, we have to also think to which planet and which kind of process we could find there. So this is just a list. We can have karstic caves on Mars because we have evaporites, Titans, I already showed to you, subglacial caves where we have glacier bodies, sublimation caves where we have sublimation of water or CO2. Of course, a lot of volcanic processes because volcanism is quite well present in the rocky planets, in the interior of the solar system, cryovolcanism, and so on. But the problem is that at the moment we can investigate this case only looking from satellite images. And so we have only surface looking, and we can try to infer. And this is where the knowledge about surface geomorphology, karst geomorphology, is very useful, because then we can try to interpret and predict what could be underground. So imagine that you are looking to Slovenia, to a beautiful DTM, but you cannot go and see any cave. It's quite frustrating. And, but also it's very fascinating, because we can try to think, imagine what could be there. Now there is there was a paper published by Wynne et al. I'm one of the co-authors, where we defined this term. I don't like a lot of acronyms, but they decided to put this term SAPS, subsurface access points, to any potential entrance that was seen on a planetary body. Of course, the Moon and Mars are the most important ones, but just because we have very good satellite images. And if you think about Mercury, for example, which is the image on top, the pixel, we have about one pixel is about 100 meters at the moment, 50 meters maximum. So of course, we will not see features like these entrances like on the Moon and Mars. So we have different planetary bodies, different numbers of SAPs, but this makes no sense if we consider that this is just because we can see or we cannot see them, depending on the resolution. So I will give to you just quickly two examples, Mars and the Moon, because are the closer and the one that have been better studies. Mars is a planet that offers a lot. It's kind of it's more similar to Earth. We have volcanism, we have probably cars because of the presence of water in the past. We have the ice caps, so we could have subglacial caves and so on. And also it's very interesting for space agency, because caves, as we know, are very peculiar environment. We can use them for protecting us from radiations, temperature excursions. There could be water. We know very well from here on Earth. And also they could be one of the best astrobiological targets, because the situation is that now on Mars, life cannot exist on the surface because of the UV radiation, but could still exist underground. And we know that the underground is a very good habitat for life. So here you see most of the entrances are located in the volcanic region of the Tarsis, but Mars, it's one of the places where there could be karst, and actually there's been a lot of work. I would say not a lot, because there could be much more done. But we have hints that really Mars hosts some karstic region and caves. And we had all the ingredients, because in the Noachian period and Hesperian period, we had water, liquid water on the surface of Mars. During the Noachian period, it was very similar to what looks like Earth now. In the Hesperian, we had big floods events. We have evaporites, and in quite very big regions. Could be gypsum, could be other kind of sulfates. But we have also some carbonates in some craters. And we have, of course, local tectonic activity. So there are the beautiful work done by Ioni and Nadia. They focused on different regions of Mars. I can say that we need much more karstologists and people working on these topics, because we have a lot of images available. There is a lot to do. And of course, karst is focused in this kind of sulfate layer deposit and also evaporite domes. Here are some images from the work from Ioni and Nadia and Tramontana. And you see, for example, these are these light tone deposits, so stratified deposits. You see that we have dolines, very nice closed depressions, a network of the lines with crests, and of course, if we look to salt domes, probably kieserite in this case, we have even more beautiful features, because it looks like dissolution. This is in Valle Marineri, Stolin Chasma and Coprates Chasma. We have very similar features like we see in salt domes on Earth. And we can even see cave entrances and blind valleys quite clear. So there could be network, karstic networks that are waiting to be explored. Of course, it's very difficult to get a mission to these places because of the topography, of the complexity. But think about a cave, water, salt or other sulfates. These are the best ingredients to get life still there underground, to have ecosystem habitats that could still be sustaining life under the surface of Mars. Of course, there is not only karst, there are entrances on the volcanoes, as we say, the most of them are on volcanoes. Very clear entrances of lava tube-like structures following sinuous chains with very different dimensions. You see this is in Arsia Mars, about an entrance is about 40 meters wide, and this is on the south of Arsia Mons, and you see the entrance is more than 400 meters wide. So even talking about volcanic processes, we have very different, so you see this is on Hawaii, this is very, very similar. You can even see on the surface the path. So we can have very different processes of lava tube formation. This is something that planetary geology is kind of pushing a little bit forward, where we could have different surface expression to the formation of lava tubes due to overcrusting, inflation in different kind of lava flows, or deeper inflation, deeper formation of tubes by thermal erosion, and so on. So this is some nice examples. On Mars, you have tubes which are, you can follow for more than 130 kilometers. They usually are very, very wide, but the roof should be probably not so high. We're talking about a big ellipticity of the conduit. And we can also follow them and follow the collapses. This is a compressed profile with all the sink of the roof of the tube, and there have been many studies now. We have hundreds of these tubes that has been mapped. And also we have tectonic caves. Tectonic caves are very frequent, very clear because of the presence of grabens, and we have the opening and formation of pits. And these caves on Earth are not so important, but on Mars, where the tectonism is involving much bigger masses than on Earth because of the lower gravity, then we could have much more and much more interesting also tectonic caves. And then we have something that we don't know. And you know, when we are talking about caves, just looking from the entrances, there is a lot that we cannot tell. We have a series of atypical, isolated pit craters, which are huge, gigantic. This is the bigger one, is Jan. You see the shadow, the illuminated wall is 350 meters deep, and we cannot see nothing about the floor. And so the question is how they form, and could be lava tubes. We don't think so. It should be something different. Could be thermocrust, could be something like you have deposits of ice underground and these are melted by the eruption. Could be dissolution of deep evaporate layers. Could be hypogenic speleogenesis on another planet. So there is still a lot of big questions. The moon, very quickly. The moon, we don't have karst, I'm sorry. We have only volcanism, but very interesting. Now, more than 200 pits have been identified, not only in volcanic region, but also in impact melts, so impact craters. They are quietly degraded. Think about that this kind of feature has some billions of years, okay? So it's not like Earth where the dynamics has been changing a lot the surface of the planet. But some of them seems to give access to caves or lava tubes or anyhow some kind of chambers underground. And they are very interesting because there is a big interest to understand, to access the subsurface of the moon, to understand how the volcanism was evolving through time, something that we cannot do looking from the surface, to understand how much volatiles we have in lavas on the moon, understand how big, if there is a lava tube, it could be. And then there is a lot of interest because of the potential utilization of these places. I'm thinking about Pajama Castle. People were going there and building to protect themselves from the enemies. And on the moon, space agencies want to protect astronauts from cosmic radiation, from temperature escalation. So there is also this idea to potentially put some lunar bases inside lava tubes. We have now quite good photogrammetric reconstruction of some of the pits. This is the biggest one, Tranquillitatis, 125 meters deep. We see that there are big continuations on the side, but we don't know nothing about that. Another one is Marius Hill pits, almost surely a lava tube collapse. Also, we have kind of tunnels on the side. And we have also other data that tells us there are big voids like gravimetry data. And we know that because the gravity of the moon is just 1/6 of the Earth, the stability of a lava tube is completely different. So we could have much bigger voids, much more stable voids with thinner roofs. And actually what we see in terms of the lunar rills could be just collapsed tubes. Imagine the fact that we had a lot of impacts on the moon's surface. So of course this could be roofed before and then they collapsed just 2-3 billions of years ago. But something could be there still stable. So you see a tunnel with a roof with a 1.5 kilometers wide tunnel could have a roof of only 10 meters of thickness still stable, which is quite, of course, an extreme, but it's telling something that we have to think completely different. This is a study where we put together morphometric data of lava tubes on the three planetary bodies, the Earth, Mars, and the Moon. So basically, we took all the collapses, we measured hundreds of collapses of lava tube type, let's say, of morphology, and we saw that the dimension of lava tubes change with the planetary parameters. So if a tube on Earth, usually it's about let's say, 10 to 20, maximum 40 meters in diameter. If we go to Mars, we reach 1 order of magnitude more, to 400 meters of diameter. On the Moon, we can go even more. We could have tubes that could be more than 1 kilometer in diameter. Of course, we have to go there. It's just a hypothesis, and it would be very important to see those things. So there is a big interest about CASE, about everything that is underground, so even about cars, I would say, from space agencies. And I think it's important that the cars community, the CASE community, will start to be more active and toward space agencies, because otherwise we leave our ground to astronomers. I have nothing against the astronomers, but to people that have not the experience on the ground of what CASE and cars look like. And there are ideas of mission. NASA has this moon diver mission that was proposed but not yet funded to a lunar pit, Tranquillitatis. There is studies from the European Space Agency with a big study that was done 2 years ago about a mission to the Marius Hill pit. And that could be funded in the future, and could be a mission that in 10 years could try to get a probe inside one of these pits. They're asking to inputs from the community. We have to develop new technology. But let's say that the interest is also because we are going back to the moon in 2026, 27 with the Artemis program from NASA and ESA. But what we can show to the public that is different from what we have already seen from Apollo missions. And CASE is one of those things. We want to show and have a look inside this case. For Mars, there is not really a mission planned. There are some studies by NIAC studies where also Penny Boston was involved. But let's say that for Mars, there are huge new possibilities. You know about the Perseverance rover, you know about Ingenuity drone, which is still flying on the surface of Mars, and this will allow to really have a look inside Martian caves, or even to reach areas of the Martian surface where we had cars in the past, and it could be extremely interesting also for astrobiology. So, to finish, I posted these 2 pictures. So you see here 6 astronauts from 5 different space agencies in Scotian Cave in 2019. So during the CASE training of the European Space Agency, the European Space Agency has been training now more than 35 astronauts from all the main space agencies. And we are coming here now to Slovenia to train them. Of course, the training is not just to train them to explore a karstic case on Mars, but it's more a behavioral training, exploration training. But it's just to say that where Marinich, Muller were going to explore now, there are astronauts that are going, and from there we hope that they will go to the moon and farther. And this picture is from the last conference in Lanzarote. We gathered more than 100 scientists from different fields in Lanzarote to talk about planetary caves exploration, to push for exploring caves on other planets, and to attract the attention again of space agencies to convince them that it's worth to go farther and to really know something about the subsurface of other planets rather than just on Earth. Thank you.
Thanks for presenting us. Always interesting theme. For sure, there are some questions. One quick question.
So I'm curious, in comparing the karst-like landscapes on Earth, on Mars, on Titan, is there much we know about how similar or different these landscapes look? Well, you know, what's different about them?
I mean, there is still a lot to study. There are big differences, of course, in dimension of the features. I mean, the only two cases now, actually extraterrestrial cases of really karst, are Mars, the one studied by Nadia, and Titan. In Titan, for example, the dimension of the feature is much bigger than on Earth. In Mars, I think more or less the IPs are bigger probably than terrestrial ones, but more or less the features are kind of similar. So, but there is a lot to do in terms of morphometry to really understand. For example, just for the lava tubes, people were just saying there are lava tubes on Mars, on the moon, and we have entrances, and you see the features, but nobody was going there really to measure the dimension, and we discovered that it's completely different from one planet to another. So there is a lot to do in terms of studying further morphometry and then modeling the processes that could be quite different.
Okay, thank you very much. We'll move to the last presentation of this year's school. It will be given by Magda. 10, 15 minutes. You will still have time for lunch, don't worry.
Okay, hello everybody. I would like for the closure to present the latest outcome result of our research. I will bring you in the digital world now. And I would like to present the multidisciplinary metadata portal we prepare at the Institute. So believe me, on my word, this is the product of the Karst Research Institute Data Center, who was established based on the free research infrastructures. Thank you. Environmental research infrastructures on which our institute is the headquarters in Slovenia and the leading partner. Those free research infrastructures are LifeWatch, EPOS, ERIC, and ELTER-RI. All three are based on environmental research, and in 2019, to give you a little background, two of the infrastructures, LifeWatch and EPOS, they were granted from the Slovenia Ministry of Research and Science and Sport and the European Commission through a grant that helped us to establish the data center and also to acquire new research equipment. Having new research equipment is meaning that we have more data. So this data should be organized, harvested in a proper way, managed in a proper way. And that's why we were looking for a platform to present better our data, being the new ones or the legacy data we have at the Institute. So we look at the GeoNetwork, an open source platform, who was helping us to cataloging our data. So GeoNetwork, it's a catalog application helping to manage spatially reference resources and provide with metadata editing, and search functions. Has also a map viewer, has numerous, the platform is used by numerous initiatives across the world. Also has interoperability between different types of projects and metadata standards. It's easy to use. It's a friendly platform. Has search options for the data and meaningful resources to provide, and so on. The metadata portal, you can even type if you have access at the internet, is the link up in the right side. We started to build it between 2021 and 2022. Actually, the work is still in progress because it's every time something to improve. We build a system, we build this platform with the help of our new collaborator, Jean Cafol, who is our DJ at the conference, let's say. And he was doing the technicality behind the portal. The architecture is compliant with fair data principle, findability, accessibility, interoperability, and reuse of the data. We have, of course, we didn't invent the wheel, but we have similar platforms at the sister projects or at the mother projects. LifeWatch, Eric has also similar metadata portal who help us to get intuitive on our data. Also, there is a Slovenian-inspired metadata portal who is also cataloging different spatial resources. We have built here a series of resources according with our needs at the Institute and our research. As you know, Karst has quite a multidisciplinary profile, so we need to accommodate as much as possible file types and resources. So we have data sets, we have media files, we have research equipment, also a catalog of research equipment, or catalog of research sites, virtual labs, services and databases, workflows and models. This list actually could be developed further according with future needs. Why it's important, this catalog? Because it's the base for our Karst database, who is also in preparation, and hopefully we can have more a better base to build it. And also our catalog provides a temporal and spatial link between specific sites and their data as part of these free projects who were actually helping to build this catalog. Related with this portal, we have also a cloud repository, because we need a place to not only to show the metadata, we need also a place to store the data. So with the help of these projects, we managed to build our own cloud. We have virtually unlimited capacity, but technically, we have around 150 terabytes shared between two main projects, LifeWatch and EPOS. Also, the cloud is easy to use, has also desktop and mobile clients, supports synchronization, also have group folders for collaboration, and what is important for researchers, also have this collaborative document editing, similar like using Google documents, let's say. There are benefits to use such catalog because in the catalog there are standardized metadata for multidisciplinary research domains. Let's say in Karst we have hydrological, biogeochemical data,
time series, we have models, we have codes. We need to accommodate all these kind of resources. Another benefit is that there is an established platform with interoperability APIs. Users who is using actually our portal, besides our institute, we give access to our collaborators in the free consortia of the LifeWatch, EPOS, and Delta Slovenia. And of course, we have other stakeholders and collaborators from Slovenia and abroad who can use the platform to register their own resources or to, if we have collaborative projects, to include the data from these projects. As I said, it's a product, it's an outcome of these research infrastructures where projects we have at the institute, but without the whole institute and our researchers to commit to this effort, we don't expect to grow. So that's why we count, first of all, on our collaborators from the Institute to grow this portal, and further to our external partners. And because I don't want to be only dry words, we want to show you a little how it's functioning, this portal. So I don't need to click anymore. So the portal is in English only. Has the metadata, to include your metadata in the portal, you need to have a sign-in account. For this, you can just request once you want to work with us. You need to get approved and to get a sign-in account, even for publishing, even just for basic editing. Metadata and templates from the portal are accommodating metadata standards for the geographical and geological data like ISO, Dublin Core, which is a more general metadata standard which covers most of the data. We have also in progress email GBIF for Darwin Core. This accommodates biodiversity data, which gives you also templates for including taxonomy in the portal. Otherwise, the portal doesn't have native such template. Okay. What kind of resources? Once again, so we have data sets. Here we include several templates. Actually, we create templates to help our colleagues to easily include their data. So we have from Excel data to GIS files like shapefile. We have AutoCAD files, we have templates for seismological data, which are also specific and also from the EPOS project. We have tabular data in general. So we just, we will show after how to fill up such. We have also photo and video. Okay, so we have also photo and video collections more dedicated to media. We have research equipment. It's a list of most recent research equipment we acquired through these research infrastructures. I think for the partners, it's important to see the collaborative environment at the Institute. So we use this platform, this catalog also as a visit card for our research. We have also services and databases. We accommodate several databases from our colleagues from the Biology Institute at the Zöldös Zoo. We also have a bird ringing database we accommodate. We have actually on our servers at the Institute. We have 2 virtual labs quite a product of LifeWatch Slovenia. If you click on. So we have, okay, we'll show it later. And the last one, no. And we have also models and codes. This, just to visualize the portal. Yeah, these are very specific for our institute. So we have a virtual laboratory in progress to be developed, Karst groundwater habitat and Proteus Watch, both made in collaboration with Tular Cave Laboratory. And also we have models and codes, maybe also to show, because in karstology, in speleogenesis, it's important to show also how actually goes the modeling. So we have several already published. As another overview, maybe just to see how you add actually a data set. You still have some time, maybe. So we choose an example to add an Excel file, all the fields marked with stars we need to fill up. So we can add, let's say, some measurement data of temperature, of climatic data. You need to add an abstract, something to describing the data, a publication, a related project, what kind of equipment. As much as possible, information is very welcome. Of course, you give the contact person or the author of this data. It could be several, or it could be only one. If you go further down, it's a very important part is to add the keywords. The more keywords you add, the more findable you have in the portal. So be as more explicit as possible and add keywords related with the locality, with the type of measurements, with the projects and so on. And also to be easy, if you don't want to type everything, you can include this file in some certain categories already established in the background. So you can choose from the categories if they are part of the project, if part of the Karst database, and you mark all the options. They are here listed. Okay. You can add also some geographical extent. I forgot to say also the portal has a map viewer. It's not so precise, so you can only draw an extent, like a polygon, but we also recommend to add in the data the coordinates. So once it's just informative where the data are, from where the data are extracted. Also, you can add if data are already attached here to the metadata portal or they are available by request. This depends on researcher how they want to share this data. Usually, all these data are stored in the cloud repository, and it's easy just to map a link to the metadata. And save and close, and this is the and then publish when you are ready with all the data. You can add also a photograph to relevant to this data and other resources. You can add also a parent link, a parent file. Let's say these data are part of the specific research sites. They are data from a cave, which is already registered. So you put link to a parent and just search here, let's say, Piuca, in the upper Piuca Valley, let's say. And this will be like a sibling for this research site. Okay, you can save and close. And later, when you're ready, you can just make it publicly. We have also, we build also a help customized for this portal. It's already in the, so it's available if you, it's actually very explicit how to add data, how to use the portal. It's still to add for, this is for only for data sets, but once you know this part, it's easy for every kind of resources to add. So this is my presentation. Thank you. I hope you will use and benefit from our portal.
Thanks, Magda, also to present us this important part of the databases we are establishing. We have one short question, if needed. If not, you have quite a lot of contacts, even Magda and Jean here, for additional information. Don't miss the departure in less than 2 hours in front of the bus station for those who are going to the fieldwork. And thanks everybody, to those who contributed with the lectures, and also to you others attendees. Thank you.
Will you be strict with the bus time today as well?
For sure.
Since we were late.
But you have more than almost half an hour more than yesterday. Yeah, because yesterday it was 1.5 hours. Yeah. We are finishing with oral part of the Karstological school, so with presentations. We will stop live streaming now, and we will move to the fieldwork. Thank everybody who participating online, and see you hopefully on the next Karstological school next year.
It's nothing bad. It's just I use